Okay, good afternoon. Welcome to the second day of the Behavioral and Social Sciences Research Festival. I'm Wendy Smith. I'm the Associate Director for the Office of Behavioral and Social Sciences Research here at NIH. I want to take a minute to thank our speakers from yesterday for their excellent presentations. If you weren't able to join us yesterday, I just want to let you know that the entire festival is being recorded and it will be archived on the NIH OBSSR website, and it should be available in a couple months. So feel free to check back for that information. Also, please note that the chat option is disabled. So if you do have questions for our speakers this afternoon, use the Q&A function that you'll see at the bottom of your screen. Really appreciate that. Thank you. I now have the great pleasure to introduce our keynote speaker, Dr. Richard Wycheck. Dr. Wycheck is the director of the National Institute on Environmental Health Sciences and the National Toxicology Program. He was named in 2020 after serving as the deputy of that institute since 2011. He has as I looked at his bio sketch, he has a very long and distinguished career. I think we can put a link perhaps in the chat for some of the specifics.
Make it short.
I can make it short. I think we'll put a chat, but some of the highlights. He's a molecular geneticist with a doctorate in molecular biology from Case Western, did postdoctoral training at Harvard Medical School. He's been at the Oak Ridge National Laboratory. These are all very senior leadership positions. Director of the Office of Functional Genomics. He was the vice chair for research and a professor at the Department of Pediatrics at Case Western Reserve University. He served as the head at the Parke-Davis Laboratory for Molecular Genetics, the chief scientific officer at Lynx Therapeutics, and just before coming to NIH, he was the president and the CEO of the Jackson Laboratory. And I'm sure there's many more details, but that was my condensed version of this really remarkable career. He'll be speaking on the role of behavioral and social sciences and environmental health, and talking about some of the opportunities for integration and collaboration. So I will pass this now to Dr. Wycheck.
Terrific. Well, thank you, Wendy. I just want to double check, make sure the microphone is working. Everyone can hear me okay? Uh-oh. That's not good.
Yes, you sound great.
Okay, yeah. Good. Well, it's great to be here, and I want to thank Drs. Hunter and Smith for this opportunity to speak to all of you. I thought I'd take advantage of this opportunity to provide an overview of the type of work that we do at NIEHS, and I'm going to very specifically talk about how those behavioral and social science approaches are integral to what we do to achieve our mission. So let's go to the next slide, please. So what differentiates NIEHS from the 26 other institutes and centers across the NIH are our mission and our vision. So as you can see on the slide here, the mission is to discover how the environment affects people in order to promote healthier lives. And our vision is to provide global leadership for innovative research that improves public health by preventing disease and disability. So the two elements of our vision that I want to emphasize are the innovative part and the prevention part. So we aim to be promoting the type of innovative thinking that will keep the environmental health sciences community at the cutting edge of advancing human health. And most importantly, the primary focus of NIEHS is on the prevention of adverse health effects. And we aim to do this by carefully studying the environment to identify factors that can either negatively or positively impact human health. Next slide, please. Now, we defined the environment very broadly, and it includes a whole variety of different environmental exposures that can influence human health. So we study the types of environmental exposures that fall under the 4 categories that are listed on this slide, shown in different boxes. So exposures in the category of physical and chemical include agents like pollutants in the air, soil, and the water, pesticides, other agricultural chemicals, synthetic chemicals, for example, like PFAS, which has been in the news recently. This is the chemical compounds in things like Teflon and Scotchgard. We also study a variety of exposures under the social category, which would include factors, for example, health disparities, psychosocial stress, and structural racism. In the ecosystem category, we study exposures related to the built environment, things like the population density, land use, and green and blue space. And finally, in the last category being lifestyle, we study factors that relate to exercise, for example, sleep, nutrition, and smoking. So the bottom line is that we are interested in how a broad assortment of different environmental exposures can positively or negatively impact our health. But what we have come to discover is that if we truly want to understand the impact of the environment, we have to embrace what's called the exposome framework, and you're seeing that on this slide. It's the totality of exposures over the life course. So in other words, we have to factor all of these exposures into the way that we conduct our research. And hopefully you will see we fundamentally embrace the approaches used in behavioral and social science research to achieve our goals. And I'll spend the rest of my presentation describing a number of behavioral and social science experiments that we're doing and supporting in support of this exposome framework. Next slide, please. So in much the same way that chemical exposures are toxins, the environmental health sciences community now recognizes and really embraces the fact that racism is a toxin to human health. So social structures based on race determine differential access to opportunities, risks, and resources that drive health. Discrimination has been linked to adverse mental and physical health outcomes. Segregated areas which lack quality schools, good jobs, safe housing and playgrounds, healthcare, transportation, a clean environment are all drivers for racial health disparities. The physical and social conditions that characterize segregated areas lead to elevated exposure to chemical hazards and to psychosocial stressors. And NIEHS has been working to study the impacts of these environmental influences in order to ultimately understand their impacts on human health. Next slide, please. And we're coming to realize that the historical practice of redlining, which is where the federal government created maps that rate the riskiness of different neighborhoods for real estate investment, redlining continues to influence present-day disparities in health. Race played a defining role in this redlining. Black and immigrant neighborhoods were typically rated hazardous and were outlined in red, hence the name redlining. Being a red-line district denoted a perilous place to lend money, according to the federal government. By contrast, those white neighborhoods were often outlined in blue and green and were subsequently favored for investment. So studies supported by NIEHS have shown that people living in red-line neighborhoods have adverse birth outcomes. They have increased poverty, higher exposure to diesel emissions, and they have an increased rate of asthma-related emergency room visits. Next slide, please. And the historical redlining also resulted in certain communities having reduced access to environmental factors that are beneficial for health. For example, historically redlined neighborhoods have fewer trees and less green space. And we know from work supported by NIEHS that green space is good for health and the environment. For example, we have funded investigators that have shown that people living in neighborhoods with high levels of green space had decreased exposure to volatile organic compounds, and they have decreased levels of biomarkers associated with heart disease. Next slide. And under the exposome framework, I also want to point out that sleep is a lifestyle factor that can affect many aspects of health. Health. So Dr. Chandra Jackson in our Division of Intramural Research studies how attributes of the neighborhood, housing, and work conditions can ultimately affect sleep health and cardiometabolic dysfunction. So she's determined that communities of color and socioeconomically disadvantaged groups are disproportionately exposed to factors that disrupt sleep. So these factors include increased light, noise, poor air quality, and psychosocial stress. So her studies have found that blacks have a higher prevalence of both poor sleep and cardiometabolic diseases when compared to whites. But when blacks and whites live in comparable conditions, these disparities disappear. So Chandra and her colleagues found that blacks and whites living, for example, in government-assisted rental housing, also known as this public housing, showed no differences in sleep duration. However, those in unassisted housing, blacks got fewer hours of sleep compared to whites. So overall, by examining the social, cultural, and physical environments leading to sleep deficiencies, we're now in a much better position to identify those factors that can reduce sleep disturbances and improve health. Next slide, please. And we are also examining the impact of structural racism on agricultural work health. So using an RFA that we issued, which is entitled Understanding and Addressing the Impact of Structural Racism and Discrimination on Minority Health and Health Disparities, we are supporting work to assess the mechanisms by which structural racism shapes COVID-19 trends among Black and Latinx, migrant, and seasonal farm workers. Many of these individuals live in economically segregated communities with substandard and crowded housing conditions with unsafe or limited water supply. The research that we're supporting will take a multidisciplinary approach. It's where we integrate environmental health sciences, community-based participatory research, statistical analysis, and public health approaches that include legal analysis. We expect that results from these studies will inform the design of policies and best practices to counter long-standing mechanisms of structural racism impacting migrant and seasoned farm workers. And we also hope that this will optimize living and working conditions for members of these communities and provide better health protections and help control future outbreaks of infectious diseases among these vulnerable workers. Next slide, please. So now I want to provide a few examples of how behavioral and social science research can advance environmental health sciences. So these examples present opportunities for stronger collaborations between OBSSR and for the incorporation of BSSR approaches into the work that we support at NIEHS. Next slide. So this is a graphic that shows how the social determinants of health, such as economic stability, education and healthcare access and quality, social and community context, and neighborhood, and the built environment interface with the exposomic framework that I used, that I explained just a few slides ago, that we use to study the environment at NIEHS. So I feel strongly that environmental health scientists must use behavioral and social science research, implementation science, and community-engaged research to address the complex problems that lie at the interface, the intersection between social determinants of health and the issues related to environmental health. Next slide, please. So this slide lists a few approaches environmental health science researchers use to gain insight into community perspectives and lived experiences that shape their knowledge, attitudes, and beliefs about their environment. So we found that oral histories offer community perspectives and concerns about exposures. And these oral histories are very important. Many of you may remember that this was a topic of discussion at the recent Environmental Health Disparities Center workshop that was sponsored by NIEHS, NICHD, and NIMHD. So also we're finding that tribal ecological knowledge provides a more complete understanding of the environmental and social determinants of health experienced by tribal communities. And we found that photo voice is an approach that allows participants, especially youth, to document the disparities in their environment. Next slide, please. So community-based participatory research, or of course CBPR, is a research approach commonly used by researchers supported by NIEHS. So in CBPR, community members are involved throughout the entire research process, from identifying the research questions and collecting the data to disseminating the research results. So in one project supported by NIEHS, researchers worked with immigrant Latinx cleaners in New York City to understand their knowledge and attitudes about chemicals and cleaning products. Now, interestingly, although nearly all of the cleaners agreed that chemicals in cleaning products can adversely affect health, more than half, about 52%, still believed it was necessary to use these products that contain strong chemicals, like bleach. And they wanted to use this for their work. So the researchers set up a test bathroom in the lab, and the purpose was to observe the different cleaning approaches. So in this case, some individuals would spray a product one time, whereas another person would spray a product 4 times for the same surface. So this approach provided controlled observations of how cleaning behaviors and habits differ among cleaners. So most importantly, research like this will provide survey results that will ultimately be used to develop a campaign to educate cleaners about safer alternatives, and most importantly, how they can reduce their exposure to harmful chemicals in cleaning products that they may be using for their job. Next slide. So I also want to point out that a key goal of the second theme of the NIEHS strategic plan has a focus on evidence-based prevention and intervention to reduce and avoid environmental exposures and their health impacts. So for this reason, we are building capacity in the field of implementation science. So implementation science, as I expect most of you know, is the study of methods to promote the uptake, scale up, and spread of evidence-based practices, interventions, and policies to improve public health. So we understand that OBSSR encourages implementation research to foster the uptake of effective behavioral and social interventions. Now, earlier this year, OBSSR played a key role in organizing a workshop that is helping NIEHS promote and advance the use of implementation science in environmental health science research. The workshop brought together environmental health and implementation scientists to discuss how they can work together to improve environmental health equity. The workshop featured discussions focused on 4 areas where the fields of environmental health sciences and implementation science come together. And these were environmental health disparities and environmental justice, prevention and interventions, climate change and disasters, and community engaged research. Next slide, please. So one area where the environmental health science and implementation science fields intersect is on studies that relate to the kind of household air pollution that is generated by burning of solid fuels for cooking. The Clean Cooking Implementation Science Network was a partnership designed to support the use of implementation science to improve the uptake of sustained use of clean fuels with a focus on how to reduce the millions of deaths caused each year by exposure to household air pollution. NIEHS staff participated in this partnership, and this partnership involved the Fogarty International Center, the NIH Common Fund, other institutes and centers across the NIH, USAID, CDC, EPA, and the Global Alliance for Clean Cooking. So overall, the initiative highlighted that there are multiple barriers to the use of clean fuels like LPG. And the fact is that the use of LPG can reduce the millions of deaths every year caused by indoor air pollution. And these barriers span the individual, the community, societal, and policy levels. This work also revealed that there are multi-level interventions that are needed. Next slide. So in the context of the barriers and facilitators to adoption of LPG for cleaner cooking practices in India, one study supported by NIEHS identified several factors that impacted adoption of cleaner fuel. The first of these was affordability. Income is a significant determinant in India. It was clear that there's a greater adoption of LPG in higher income groups. So the second is accessibility. What is readily available in rural areas, and women collect biomass and prioritize it over alternatives because it's free and it's readily accessible. So the third is awareness. Researchers found that adoption of LPG is impacted by concerns about its safety and explosiveness. So therefore, in the end, awareness campaigns were found to be a key component to support LPG adoption, especially in poor households. Next slide. So NIEHS also encourages the translation of environmental health research from the lab and moving it into the real world through the development of evidence-based strategies that protect and improve human health. So NIEHS supports basic research using cell and rodent-based studies that have linked PCB exposure to increased oxidative stress, inflammation, and heart disease. So the research team that conducted this basic research subsequently went on to find that compounds in fruits and vegetables, this is nutrition, these compounds such as inulin and flavonoids can protect against PCB toxicity. So based on this finding, they implemented an educational program to increase fruit and vegetable consumption among people exposed to PCBs. So many of these interventions focused on older adults since they may be, they're clearly more susceptible to the hazardous exposures like PCBs. So in the end, it's remarkable that participants in this educational effort reported that they made positive behavioral changes, including eating more fruits and vegetables, reading food labels, and consuming fewer sugar-sweetened drinks and consuming less fast food. Next slide. So another example of the work that NIEHS supports in translation is a new program called the Collaborative Centers for Children's Environmental Health Research and Translation. The new center is dedicated to the translation of research that can improve children's health. The program builds off of more than 20 years of NIEHS-funded research that has provided insights into how the environment affects children's health. So NIEHS staff and stakeholders saw a need to translate this accumulated knowledge of this accumulated knowledge and to really bring it to affect children's health. And they're doing this through the development of translational tools, approaches, and a variety of different interventions. The program is made up of 6 centers across the country that bring together the collective expertise of the environmental health sciences community, communication, behavioral and social sciences, implementation science, medicine, and policymakers. An example of this multidisciplinary collaboration is a center at Oregon State University, and it's called ASPIRE, the ASPIRE Center, which is short for Advancing Science, Practice Programming, and Policy and Research Translation for Children's Environmental Health. The center is co-led by an environmental health science researcher and a developmental psychologist. So together they bring the expertise both in children's development, behavior, and learning, as well as dissemination strategies for real-world environments, with an example being early childhood centers. So next slide, please. So NIHS is also supporting a T32 grant at Northeastern University that works to bring BSSR skills into the environmental health sciences. This is a partnership with the Silent Spring Institute, which is a science-based environmental nonprofit organization. The program involves work with community organization, and it prepares doctoral and postdoctoral trainees to operate at the intersection of environmental health and social sciences. Next slide. So NIHS is also very interested in building capacity to conduct community-engaged research. For example, we are co-funding an R25 with OBSSR, thank you. And this R25 aims to address the need to use innovative and community-engaged methodologies in the behavioral and social sciences. The award supports a national training program that aims to enhance the capability of BSSR researchers to use community-based participatory research to ultimately examine and address the health equities. The program begins with a 1-week intensive course with hands-on learning experiences that pairs researchers with community partners. The program also includes mentoring and career development activities, a train-the-trainers model to build capacity and extend program outreach, dissemination of the knowledge gained, sharing of the curriculum, and a focus on increasing diversity in the BSSR workforce. Next slide, please. So the R25 that I just described, it's been a real success story. But let me now turn to describing work in the behavioral and social sciences that are part of a new emerging program at the NIH for studying the health consequences of climate change. Next slide. Okay, so to introduce the topic of climate change, you may all recall that it was back in 2021, soon after the inauguration of President Biden, that the President issued Executive Order 14008. And he pointed to the NIH to take on the challenges of addressing the health effects associated with climate change. And the NIH was ready to take on this challenge because we've been studying the health effects of climate change at a modest level for quite a number of years. This was especially true for NIHS. But we recognized that additional money could help to catalyze a much larger effort to undertake these challenges associated with the health effects of climate change. So therefore, we are engaged. We were engaged, working together, and we're actually quite encouraged to learn that Congress and the President have put anywhere from $10 million to $100 million in their budgets that would clearly be dedicated to studying and catalyzing these additional experiments to deal with the health effects of climate change. So we're encouraged that this Congress is making this a priority for FY23. But as we learned last year, we're not really quite certain that we will ultimately be appropriated this money, but nevertheless, we've started working to develop an NIH-wide strategy for climate change and health. And we're doing this on an NIH-wide scale because it's clear that this is a topic that's much bigger than what any single IC can accomplish on its own. And it's also clear that there are many institutes and centers at the NIH that have both capabilities and a keen interest in this area of research. So therefore, I've taken the initiative to reach out to Dr. Bianchi at CHD, Dr. Gibbons at NHLBI, Dr. Glass at Fogarty, Dr. Gordon at Mental Health, Dr. Perestable at NIMHD, and Dr. Zank at the Nursing Research Institute to form what we are calling the Executive Committee on Climate Change and Health. So together we have re-energized a long-standing working group with over 100 members from across the NIH. The working group is co-chaired by Aubrey Miller at NIEHS and Josh Rosenthal at the Fogarty International Center. So our own Gwen Coleman at NIEHS is providing the much-needed central coordination to keep the vast number of moving parts all working in synchrony with each other. So the steering committee is, there's also a steering committee that that Josh and Aubrey and Gwen put together, and this is it has membership from the ICs that are represented on the executive committee. I mean, the steering committee is working with Drs. Miller and Rosenthal to make sure that the goals of the working group are achieved. And the working group has been busy, very busy on many projects, including most notably the development of this NIH-wide strategic framework for climate change and health. Next slide, please. I just want to talk about climate change. This slide illustrates the enormous complexity associated with how climate change impacts health outcomes. So climate drivers at the top affect health outcomes either directly through weather events such as extreme heat, wildfires, droughts, storm surges, and floods, or indirectly through a series of exposure pathways such as air and water quality, food quality, infectious diseases, and massive population displacement events. So these pathways are themselves influenced by environmental context related to land use, geography, infrastructure, and agriculture. And it's also clear that these pathways are also influenced by the social and behavioral contexts shown on the right here, involving age and gender, race and ethnicity, poverty, housing and infrastructure, education, discrimination, and access to health care. So therefore, any approach to address the health impacts of climate change, we absolutely need to involve innovative transdisciplinary and systems-based approaches. Next slide. And what will be important in any plan to execute on the health effects of climate change will be the recognition that climate change doesn't affect everyone equally. So we need to recognize that under-resourced and marginalized populations, like some communities of color, populations with low income and low educational attainment, as well as immigrant groups, including those with limited English proficiency, many indigenous populations, exposed farm and construction workers, and persons with disabilities, these are all especially impacted by climate change. And we also need to recognize that the fetus, infants, young children, pregnant women, the elderly, and others in vulnerable life stages are also, also those with chronic medical conditions are especially impacted by the climate change. And we have to, in the lower right-hand corners, recognize that the populations in lower and middle income countries are especially at increased risk from the impact of climate change due to the higher rates of existing diseases, malnutrition, and extreme poverty. Next slide, please. So the strategic framework that we've put together was ultimately developed using several sources of input data, which included a portfolio analysis by the working group, a strategic whiteboarding session, a landscape analysis, and over 180 substantive responses from an RFI that the working group issued back in August of 2021. The framework is illustrated with this centerpiece diagram. We call it the COG diagram for obvious reasons. So in brief, the framework is based on 4 different core elements shown at the center of this COG diagram. And these include health effects research, health equity, training and capacity building, and intervention science. And at the center is a focus on multidisciplinary transformative research. The work will be conducted with global cross-sector, interagency, and community partnerships. And the cogs on the circumference are meant to illustrate specific types of science that will need to be conducted to support the framework. And as you can see, I'm not going to go through all of these, but you can see that there are research areas that are truly multidisciplinary in nature, and they range from basic mechanistic research, epidemiology and predictive modeling, adaptation research, and of course, behavioral and social science research. We developed a website, so if you're interested, take a look, nih.gov/climate and health. It's one word. And stay tuned. We are hoping to get that appropriation from Congress, and hopefully we can put some of that money to use to help to catalyze these innovative new projects addressing the health effects of climate change. Next slide, please. I'd like to give you now several examples of the social and behavioral sciences as it relates to addressing the health effects of climate change. So the first example is related, well, it's a project that's co-funded by NIEHS and OBSSR. Again, thank you. So in this case, researchers are evaluating the impacts of home weatherization programs on the health of older adults. So older adults are especially vulnerable to extreme temperatures, and many live in homes that do not provide adequate protection from temperature extremes. So specifically, researchers are assessing how measures of weatherization impact cognitive function, sleep quality, sleep duration, and financial worry. Focus of the work will be to monetize the health benefits or the health impacts of weatherization measures, and we hope that this will improve the health of older adults. We also expect that this research will help inform policies for energy efficiency and weatherization subsidies. Next slide, please. And we've also found that children are especially sensitive to extreme temperatures, and this sensitivity may vary by age in ways that have very important implications for public health. Therefore, we are supporting a project to examine the variation in children's susceptibility to heat, and we are doing this by studying multiple health outcomes by age across New York State, including New York City. The investigators involved in this research are considering social and structural factors that may worsen or mitigate heat impacts on child health. And this would include impact on poverty rates and urban density. The study findings will help to inform heat alert programs and disseminate research-informed messages to local health departments and clinicians serving children. So this project will help to improve resource allocation to protect the most vulnerable by identifying those populations which require better heat-related prevention services. Next slide. And I think we recognize climate change also impacts our mental health. In fact, studies show that more than 2/3 of young people are worried about climate change and how it will affect their future. So therefore, we are supporting a study at NIEHS to leverage data from the Crisis Text Line to examine the effects of hurricanes, wildfires, and heat waves on youth mental health outcomes. Included in the outcomes are suicidal thoughts, self-harm, anxiety and stress, depression, and substance abuse. So researchers involved in this work also aim to identify the individual and community level factors like age, gender identity, race, ethnicity, poverty, racial segregation, and urban greenness that promote mental health resilience in youth that are impacted by a climate disaster. So the expectation is that these studies will generate data that can be used to help to inform crisis response and mental health training efforts. So next slide, I'm winding up here. I want to make sure that we have plenty of time for Q&A. So in sum, hopefully you have a really good idea of what we're doing. And so hopefully it's clear to you that a good part of our research portfolio involves transdisciplinary approaches that involve environmental health scientists, sociologists, anthropologists, historians, data scientists, economists, communication researchers, and policy researchers all working together. The issues we study in the environmental health sciences are complex, and it's absolutely necessary to engage in this collaborative environment to make progress to improve public health. Okay, so let's go to the next slide. I think that's the last. Yep. So thank you all for this opportunity, and I am happy. Why don't we turn the slides off? And I'm happy to entertain any and all questions you may have. And I noticed that my colleague, Dr. Martin, Lindsay Martin, is on. So if there are any questions that I need some help on, Lindsay and maybe Liam O'Fallon. I don't know if Liam is on, but we'll be happy to take those questions.
Thank you so much, Dr. Wycheck. That was quite breadth and depth on all the activities at NIEHS that are certainly relevant to our mission, as well as many other across NIH. It's fascinating to hear the variety and the transdisciplinary nature of a lot of these activities. So
OBSSR has been a great partner with us, and we look forward to increasing that partnership.
Thank you. Looking to see if there's any specific questions at this point. Need to use the Q&A. It's going to take people a minute to that while we're waiting. I found your description of the climate change and health work especially fascinating because it highlights a lot of areas that people don't normally consider under that, and may be surprised come in under your mission.
Mhm. Well, it
There-
comes under our mission, but again, the point I want to make is that it's not just NIEHS.
Mhm.
It's really the multifaceted aspects of climate change really require the NIH as a whole, to pull together and I would say in almost an unprecedented way
Mhm.
to build these alliances to be taking on these multifacets that have to be dealt with around the health effects of climate change.
Mhm.
Then, of course, we have to be working together with other agencies of the federal government who are working on issues like, what do we do to mitigate the pollution from combustion of fossil fuels. So we
Mhm.
all need to be working together to tackle this critically important threat to human health and
Mhm.
threat to humanity on the face of the earth.
Yeah.
So it's actually truly been remarkable how other IC directors have stepped up and their willingness to engage with us. There are 6 other IC directors on the executive committee, and it's not that only 6 others were interested. Many others were interested. It's just that we found that going beyond 7 IC directors as on the executive committee would just make it unmanageable.
Mhm.
But we will be rotating other IC directors on and off as necessary. But there's just a lot of interest, and there's a lot of interest coming in from Congress as well, which is good.
It's very good. It reminds me of some of the other all hands on deck sort of programs
Exactly.
coming out of NIH to address crises in public health.
Well, we just came through one of these in COVID-19,
Exactly.
and actually that's part of my evening and weekend job. I was the co-chair of the Rapid Acceleration of Diagnostics with Dr. Schwetz, and I think that's another real feather in our cap at the NIH. The things that we were able to do in breakneck speed
Yes.
around both testing and therapeutics.
Mhm.
And a lot of that I really attribute to the remarkable leadership from Dr. Collins, and then following up with Dr. Tabak now.
We have a couple questions coming in. Are there any particular areas of environmental risk or threat wherein you think is especially important or necessary to investigate gene-environment interactions?
Well, I didn't have time to go into that. Uh, so there's an emerging new area in environmental health sciences called the precision environmental health, and that's all based on the fact that people respond to the environment in different ways. So what may be exquisitely dangerous for one person may not apply to someone else. So there's a lot of interest in these gene by environment effects. But it's not just genetics, it's also epigenetics. And what we're finding, in fact, I'm a geneticist by training, a molecular geneticist, although some of my more recent research is on epigenetics. Because what we're finding is that many of those environmental influences can modify the DNA in such a way that it affects the way that genes are being expressed. So it's this new program, Precision Environmental Health. If you're interested, you'll have to invite me back because that's a lecture in itself. But that's again another multifaceted, which involves lots of gene by environment effects.
Another question. Do you think it's relevant to record hazards of climate change for individuals in their electronic health records?
So hazards for climate change and their electronic medical records. I mean, generally speaking, I think it's critically important to get environmental exposures into electronic medical records, which I think, quite frankly, is currently is an embarrassment, because for the most part, physicians are not asking their patients, where do you live and where do you work, and what are the consequences of some of these health effects of climate change. So absolutely, we need to be factoring these into the way that we diagnosis diseases, and just having a better sense of whole person health. So absolutely, I'm a strong proponent of getting it into the electronic medical records. Does that answer the question?
Yes, I think so.
Okay, good.
Um, here's another sort of question that's helpful for our specific audience is how can we better prepare behavioral and social science researchers for this potential influx of funding on climate change health? And I'm reading that in the context of if we need these partnerships, these collaborations, these multidisciplinary sort of approaches, should people start thinking about trying to develop those kinds of relationships to better prepare for coming in for some of the kinds of things you might see coming down the road?
Well, so to answer the question, I'll answer it in many different ways. The first one is to develop the mindset that we need to be working together on things. So it's not just about OBSSR and NIEHS and individual ICs kind of getting some proportion of the money and then tearing into this and running off in siloed fashions. That's not going to work. And so what we're promoting is that this really needs to be done in a collaborative mode. And to promote the collaboration, what the working group together with the executive committee and the steering committee did was came up with these documents to define what it is that needs to get done. You'll remember on that cog diagram that I showed on the periphery are a whole bunch of different kinds of science that have to get done in support of those 4 core elements. Mm-hmm. So I guess my response would be, if you're interested and you want to start preparing, get on the website nih.gov/climate and health and read through the documents. And the working group is open to anyone who may be interested in this topic area. So if you're not currently on the working group and you want representation on the working group, let Gwen Coleman or Aubrey Miller or Josh Rosenthal or me know, and we'll make sure that you have the ability to join the working group. We ask that if you're on the working group, we're going to be asking that you roll up your sleeves and that you have some dedicated time, because this is for real. It's not just about discussing these things now, but it's coming up with plans on how can we get the NIH more directly involved in this very important area of research. Now, just another comment that I'll make is that we're hoping for that catalytic funding coming through in the FY23 budget. But the NIH has a $46 billion budget, and so we're also trying to just increase awareness across all 27 ICs that studying the health effects of climate change, should be a priority for all of the institutes and the centers and offices across the NIH. So hopefully some of those unsolicited grant applications that are coming in, hopefully more of them will be focused on the health effects of climate change, and that different ICs and offices within the NIH will look favorably upon these applications that are coming in.
Okay. And just to clarify, is this a working group because our audience is both NIH and public? So are you, for members of this working group?
Working group is exclusively members of the NIH.
Okay.
The other thing is, if you get on our website, nih.gov/climate and health, you will see that the executive committee is able to come up with some, I'll call it startup funding to support what we call a resource, the RCC, the Resource Center Core, which is the first attempt to create a community of practice around climate change and health. So if we're all going to be jumping into this and working collaboratively together, that RCC was really meant to be an attempt to get people working together, providing some of that framework so it makes it easy for us to seamlessly share our data, share our ideas, and get things moving. You'll also see that we supported the first phase of a community engagement core. Mm-hmm. And I think the executive committee absolutely recognizes, and a lot of this is based on work OBSSR, NIEHS, and others have done to recognize the importance of community engagement. Mm-hmm. And community engagement takes time. You don't just snap your fingers and all of a sudden everybody said, "Oh, you're from the government. You're here to help. Terrific, great. We'll work with you." So you've got to build that trust, and that takes time. Mm-hmm. So we made an investment to establish some fundamental efforts to get the community engagement going. But additionally, this is a very, very long response to your
It's okay.
But we also, actually it was the Office of the Director, Dr. Schwetz and Dr. Tabak, that came up with an allocation of funding that we're using to establish what we're calling as the Climate Change and Health Scholars Program.
Mm.
So that's an attempt to bring in people from the outside. So not just federal employees, but people working in academic institutions or nonprofit organizations that are personally passionate about the issue of climate change and health and have ideas that they would like to bring on campus. So we're providing this funding and this mechanism for the Scholars Program. So you can either attend virtually, or we even have the opportunity for people to spend some period of time to actually come on campus, interface with federal employees on the working group, on the steering committee, and bring their ideas forward to help refine the plans for dealing with the health effects of climate change.
Okay, so I'm hearing a call for inclusion from NIH folks to get involved. I'm hearing some opportunities for the outside people to get involved. I'm hearing some other information too for researchers who may want to start preparing collaborations to come in, and people can hear more about this through your website.
Well, the website, and I also want to call attention to the fact that the journal Lancet has recently published a commentary that I co-authored with the other 6 members of the Executive Committee for Climate Change and Health. So I would encourage anyone who's interested to take a look at that commentary.
Excellent.
And I also want to thank Aubrey Miller and Josh Rosenthal and Gwen Coleman and others for help in assembling that commentary. But it's really an attempt to start spreading the message that the issue of the health effects of climate change is here. The NIH is developing plans to respond to this, and we want people to know that this is something that, if you have ideas, put in a grant application, and just letting people know that this is a priority for the NIH to be funding.
Excellent. So reach out to us. We can keep people in the loop. There will be announcements of upcoming things. If people struggle finding that, they can reach out to your office. They can reach out to us as well. And I think at that point, although there's more questions and we'll try to get to those offline, we're going to need now to move to our next session. But Dr. Wycheck, I want to thank you again for this really excellent presentation, this overview. And I think maybe sort of a new introduction for a lot of people online to sort of see what you're leading, the breadth and depth of these activities. It's very exciting.
Well,
so
I just
thank
want
you
to
very
comment
much.
that I've got some fabulous colleagues. I mean, I have the luxury of getting up and talking about their wonderful work, but Lindsay and Liam and others, my colleagues at NIEHS, I mean, they're doing the work, and it's been a great partnership, Wendy, with you and Christine and others in OBSSR. So let's keep it up, maintain the momentum.
Yep. Thank you again. Really appreciate that.
Yep.
So now I'm going to hand this over to Dr. Erica Spots from OBSSR, who will be taking over for the next session. Thank you again.
Thanks so much, Wendy. What a great start to the second day of the Research Festival. And I think serendipitously, this session is on social determinants of health, which is a really nice follow-up to our keynote speaker. Our first speaker will be Corinne Dubé from the University of California, San Diego, and I will let her pop up her slides and get started.
Thank you so much. So today my talk will focus on intersecting dynamics, difficult conundrums, increasing racial, ethnic, gender, and sexual diversity in HIV cure-related research involving analytical treatment interruptions. So this is the paper that we'll be presenting today. And I want to give a special thanks to all my co-authors and all our amazing study participants, as well as the National Institute of Mental Health for supporting this work. So a little bit of background on HIV cure-related research and treatment interruptions. So there are two goals being pursued with cure research, either complete elimination of HIV from the body or sustained antiretroviral treatment-free suppression. And there are multiple strategies being investigated, including latency reversal agents to reawaken HIV or the opposite, block and lock, to keep it permanently silenced. Immune-based approaches, cell and gene therapy approaches, and combination approaches. But a key feature of these studies is that we need to interrupt treatment. These are called analytical treatment interruptions, and they can help us determine the efficacy of these interventions. These ATIs have risk for participants, but also the partners of participants without HIV. Globally, there's about 250 studies that have been implemented worldwide, and this represents around $330 million invested in HIV cure-related research each year. And there's several groups implementing HIV cure-related trials. So our study methods, we recruited 21 informants and completed in-depth interviews. Informants included biomedical researchers, community members, bioethicists, social behavioral scientists, and HIV care providers in the United States. The study guide was developed in close collaboration with community members, and we analyzed our data using conventional data analysis. And we extracted key considerations for implementing ATI trials in diverse communities and settings. Some of our key findings: attention must be paid to gender and power dynamics. ATI trials should be designed and implemented through the lens of intersectionality and equity. And ATI trials may have both positive or negative effects on stigma for people with HIV and their partners. Partnership dynamics should be considered and will need to bridge the fields of HIV cure-related research with HIV prevention. And social behavioral research should be mandated and funded as part of biomedical HIV cure research efforts involving extended treatment interruptions. On the right, you can see some of the key intersecting factors, including gender and sex, age and power differentials, race and ethnicity, partnership status, and whether intimate partner violence is present in the relationship and the status of partners, among others. Some of the considerations for involving women in ATI trials: this should be recognized as a matter of equity and justice, and we should seek to understand how HIV cure research fits within the context of women's lives. We should practice gender inclusivity as all stages of clinical implementation, and for participants who can become pregnant, the risk of transmitting HIV to their fetus should be minimized through robust informed consent and protections. Research teams should recognize that people may not always have control over their ability not to conceive. And when women are ATI trial participants, they should engage and refer their negative partners to receive protection measures whenever possible. When women are partners of ATI trial participants, they should be offered pre-exposure prophylaxis and other prevention measures. We should also apply a gender lens to ATI trial implementation and carefully weigh the possible social and clinical consequences for participants and their partners. Attention should be paid to gender dynamics and power imbalances for issues around disclosure and partner protections. And women should be involved as part of HIV cure research teams and protocol reviewers to guide the implementation of research. We should also involve community organizations that are well-versed in gender and sexual diversity issues. And to facilitate participation, research teams should help women overcome barriers to participation, and we should have tailored support and counseling. Here are 2 quotes from community members. We know that a person might not have disclosed to their partner, and now they may again, if we're considering this study, now this may change the dynamics of things. Women living with HIV are at high risk of having violence perpetuated upon them than a woman with a different serostatus. There's just a lot, and there's not enough trauma-informed care out there already. And I think there are a lot of women that are in situations where there's an imbalance of power in the relationship. It brings together so many colliding issues, right? When you talk about the rates of domestic violence or intimate partner violence against women with HIV, they're exponentially higher than that of the general population. And so to bring into play a partner's participation in our trial, it increases her level of vulnerability. We want to do the ethical thing and offer PrEP to a partner, but we must first evaluate the woman's ability to have an open dialogue with her partner about her participation in the clinical trial. Here's a quote from a social behavioral scientist. There's a lot of gender and sexual power dynamics that may come into play when you're talking about HIV and STIs. Anything in that realm, whether it's cure research or treatment adherence research. Even though women tend to be much more engaged in healthcare, in other words, more willing to go to the doctor to get preventive screening, they still do not experience as much HIV treatment services as men do. In general, women have more complications when it comes to sustained viral suppression. And if if you're talking willingness to be involved in an ATI study, they might reap less benefit when there's already so many challenges that they're dealing with in the first place. And if you add the element of race, African-American women, for example, pretty much are doing the worst when it comes to reaping the benefits of all these advancements in HIV treatments. So I think there's an intersectional issue of racism and also sexism when it comes to why Black women are experiencing some of these challenges. We're also talking about issues around medical mistrust and also very real healthcare discrimination when they do have access. There are problems with Black women having undiagnosed mental illness related to HIV, which is complicating their treatment outcome. There are so many things that should be thought about for all of those: disclosure, inviting partners, doing HIV testing, all of that. Here are some considerations for involving racial and ethnic minorities in ATI trial. There should be a lot of intentionality around diversity in trial design, and we should meet communities where they are. We also need to pay attention to the diversity of research staff and cultural literacy of researchers. We should also involve diverse peer mentors and counselors. We should pay attention to logistics and implement trials with racial and justice frameworks. We should also build trust with HIV care providers and invest in culturally appropriate community engagement efforts. We also need to recognize that having a detectable viral load may create heightened vulnerabilities with respect to criminalization of HIV transmission. And as a society, we have not done a really good job destigmatizing people who do not have a non-detectable HIV status. Here's a quote from a biomedical HIV cure researcher. Given where the cure field is, sometimes I certainly hear treatment interruptions or cure studies characterized as more experimental than other kinds of research. And I think that communities of color, trans individuals, and members of particularly vulnerable and underrepresented populations are particularly vulnerable to exploratory and experimental research. And that kind of lights up historical trauma from that work in the past. So I think that just requires a great deal of sensitivity and considerations as we move forward. We also need to recognize that ATI trials may have different impacts on stigma. Stigma could be increased as a result of becoming viremic and being at risk of transmitting HIV to a partner, or stigma could be reduced as a result of helping advance the search towards an HIV cure. We need to remember that stigma relates to multiple interlocking experiences of oppression, including racism, sexism, gender binarism, heterosexism, and transphobia. And we need robust community engagement and awareness to help us counteract that stigma, including tools to help people understand why treatment interruptions may be needed. Here's a quote from a community member. The stigma of coming off of one's medication in order to participate in an ATI-involved study has huge ramifications for HIV criminalization laws in this country, in the United States and globally. That's a huge, structural and systemic barrier to engaging underrepresented communities. And I'm specifically naming Black communities who already have disproportionate involvement, or should I say, disproportionately, inequitably targeted by the criminal justice system in the United States. We also need to pay attention to partnership dynamics, and research teams should adopt intersectionality and trauma-informed and healing-centered research approaches that account for different kinds of trauma and minimize the risk of possible social harms when implementing ATI trials. And participants should be empowered to evaluate trade-offs during the informed consent process. We need trauma-informed research, that the whole history, whether it's based on gender, whether it's based on race, and even in some instances, whether it's based in geography, we don't know where people have come from, and we don't know what they all walk with. There's so many different kinds of traumas, and it's not just about women, and it's not just about Black and brown people, that the diversity is what we want. The intersectionality is what we have to understand. And so in collaboration with biomedical researchers and community members, we're developing tools to help mitigate risk of HIV transmission during analytical treatment interruptions, and also developing ethical considerations. The NIH organized a meeting in 2021 around addressing challenges that limit participation in HIV trials with treatment interruptions. This is Larry Fox, Tia Morton, and with Dr. Annette Rid from the NIH Bioethics Center, we're developing a partner protection package for HIV cure-related studies involving treatment interruptions. But obviously, we need a lot more evidence as it relates to partner protections in HIV cure trials. We also implemented a study that recruited diverse HIV serodifferent partners in the United States. And we found that there's still a lot of misunderstanding in the community around undetectable is untransmittable, or U equals U. And this study also challenged the notion that only participants are implicated with ATI trials. We also need to consider partners in our trial designs. And there's a need for education around ATIs and U=U, the provision of PrEP and other HIV prevention modalities, and how and when to integrate partners of participants. We also implemented research with demographically diverse young adults living with HIV. This work was co-led with Dr. Paria Sabiri at UCSF. And we also found unmet needs for young people to participate in this research. We'll need a lot more support around HIV disclosure, and also we'll need to help young people overcome barriers to HIV prevention, including PrEP access, because the individuals that we want represented in trials are also those that are underserved when it pertains to HIV prevention services in the United States. I also would like to highlight the work of the Female Rising Through Education, Support, and Health FRESH cohort in Durban, South Africa. This initiative by the Reagan Institute leverages the HIV prevention, early treatment, and post-intervention control research continuum. And in addition to doing basic research, this program has an economic and social structural intervention for the girls and the young women. And in addition, we're adding a social behavioral program and a robust psychosocial support and healing center design to the FRESH cohort. So these findings have a lot of implications. We need more dialogue around adequate and inclusive ATI trial designs in complex relationships and contexts in underrepresented populations. We'll need to integrate behavioral and social science research in the front end instead of an afterthought, and BSSR should be on an equal footing with biomedical research and should be part of clinical trial designs. And also partner protection efforts should be prioritized to not compromise the field, increase trial diversity, and help maintain trustworthiness of research. Thank you all so very much.
Thank you so much for a really interesting talk. I've got a couple questions keyed up for you. These HIV cure trials present significant risks because they temporarily remove people from effective HIV treatment. What are your thoughts about how we can make informed consent processes more effective, and particularly with groups experiencing marginalization?
Thank you, Dr. Stirrat. That's a great question. I think there's evidence now that we need to simplify the informed consent process. The current forms have 20, sometimes even 30 pages. So we really need to help participants through their decision-making process. And also we could have a lot more tools to make the informed consent process more meaningful. So companion documents to accompany the consent or videos. So there could be a lot of innovation around informed consent. And we could also even think about reforming the informed consent process to make it more patient and participant centered. That's a great question. Thank you.
I guess I'm going to follow up with that. I'm curious. I've been on IRBs. I'm wondering if IRBs are open to that sort of simplification and both simplification and augmentation of consent forms.
That's a great question.
It's nice to think about that, but
yeah.
Do you have experience with that? Are IRBs open to that kind of thing?
Yeah, I think it would be great to engage the IRBs around this issue and also do social behavioral research with IRB members to understand what would be acceptable to them in terms of simplifying the informed consent. So I think this could be an ongoing conversation with our IRB members.
Great, thank you. Oh, sorry, my screen just changed and it confused me. So the next question is, considering the potential impacts of ATI participation related to stigma specifically, we know that U=U has been life-changing for so many people living with HIV. Did the participants express concern in this regard specifically?
Yes, so there's a lot of concerns around U=U potentially being invalidated during the course of an ATI. And there's also the viral rebound. It can be unpredictable. Every person reacts differently. So there's a lot of concern, not just on the level of stigma, but also the transmission of HIV to partners. And this is a consistent worry that we find also as part of ongoing HIV cure-related trials. So something that we should probably monitor as part of ongoing trials as well.
Thanks. And then Mike has a follow-up question. Your research is closely integrated into biomedical clinical trials. What changes have you seen in the receptivity of biomedically oriented researchers to behavioral research over time? And how do we further this?
That's a great question. I think there's been more receptivity as we show the value of the SSR and how we can work together to help advance biomedical research in a collaborative effort. And I think how we further this is we mandate it, and so it becomes part of the design and second nature. And we encourage transdisciplinary and cross-disciplinary dialogues. I think that also really helps. And having community engaged research, community members at the table to help resolve those complex issues is also really important. Thank you.
Thank you so much for a really interesting talk and discussion afterwards. At this point, we will move on to our next speaker, who is Dr. Tiffany Powell-Wiley, who she is a Statman investigator, chief of the Social Determinants of Obesity and Cardiovascular Risk Laboratory in the cardiovascular branch of the Division of Intramural Research at the National Heart, Lung, and Blood Institute at NIH. And with that, I will let you get started. Thank you.
All right, thank you so much. And thank you to the organizers for the opportunity to be here today. I'll be talking about social determinants of cardiovascular disease. These are my disclosures. And so I'll begin just by giving some background about why, particularly as cardiologists, we are interested in the social determinants of health and what we're seeing as far as trends in the effects of social determinants on cardiovascular disease. So we know from recent data put forward by our colleagues, myself and my colleagues at NCI, we looked at the effects of, or what was affecting premature cardiovascular disease mortality in the United States, looking at CDC data from 2000 to 2015. And we saw that most specifically, counties that had the highest rates of not only obesity and diabetes, but those counties that had the lowest levels of education were at the highest risk for premature cardiovascular disease mortality, meaning mortality before the age of 65. And so when we think of education levels as a social determinant, this is one clue of the role of a social determinant in the development of adverse cardiovascular events. And so there's also been data to support how cardiovascular disease is affecting the US life expectancy. We know that particularly since 2011, we've seen a slowing of cardiovascular disease mortality rates decreasing in the United States. But this looked at first comparing cardiovascular disease mortality across countries and showed that within the United States, we have 30% higher CVD mortality as compared to other high life expectancy countries. But they also showed that with simulation modeling, they compared what would be the effects on US life expectancy in models that looked at whether or not preventing any increases in drug-related deaths, as well as any increases or in maintaining cardiovascular disease mortality rate declines that we were seeing before 2011. And this simulation model really showed that if we were able to maintain cardiovascular mortality rate declines, we would actually be seeing similar trends and similar increases in life expectancy than we saw prior to 2010 or 2011. And so the implications of these types of studies really highlight that if we're really going to affect US life expectancy, we really need to focus on cardiovascular mortality. And it's not really just the cardiovascular events, it's the risk factors that go into the development of cardiovascular disease, whether it be obesity, diabetes, or hypertension. And really, we know there's a significant role for social determinants of health in contributing, again, not just to the what we're seeing in cardiovascular disease outcomes, but also in outcomes related to obesity, diabetes, and hypertension. And so the paper that I was asked to talk about, in the paper that we published this year in Circulation Research, really tried to dig into this connection between social determinants of health and cardiovascular disease. And we defined social determinants as the economic, social, or environmental and psychosocial factors that relate to where we live, work, play, and pray, but also that influence cardiovascular disease outcomes. And we wanted to not only look at what was some of the existing data around these connections between social determinants of health and cardiovascular disease, but we also wanted to better highlight what were some of the more mechanistic connections between social determinants and cardiovascular disease. And I'll talk about those in a moment. And so the first thing that we put forward in this work was really trying to highlight a framework for how social determinants lead to the development of cardiovascular disease. And within this framework, we wanted to center health equity in those populations that are most impacted by health disparities. And so we know, for instance, as our previous speaker mentioned, we have structural determinants of health that really influence what populations experience. We know that these structural determinants of health equity, whether they be socio-political or economic context, structural racism or structural discrimination, access and quality to education and healthcare, and socioeconomic factors, as well as neighborhood and environmental factors, all of those influence more intermediary determinants of health. And these are things related to social and community context, social risk, or things that relate to social needs for individuals. And these types of determinants influence the lived experience within a community, and all of which are, all of these factors really influence an individual's social position and their life experience. And ultimately, these factors lead to both psychosocial but also environmental stressors, which have biological and psychological sequelae and outcomes that ultimately lead to the development of cardiovascular disease. And so I'll just highlight a few of these factors related to built-in physical environment, as well as psychosocial factors that we know are related to the development of cardiovascular disease. And so much of the work that has looked at connecting social determinants of health to cardiovascular disease has been cross-sectional studies, but there have been some key longitudinal studies that have really highlighted how there's this connection between particularly neighborhood built and social environment and cardiovascular outcomes. We know, for instance, that racial and ethnic segregation, residential segregation, is related to incident cardiovascular disease. The walkability within an environment is related to incident diabetes and hypertension. We have done quite a bit of work in my groups looking at the relationship between neighborhood disadvantage and how that relates to obesity related outcomes, but we also know it relates to incident heart failure. But it's important to also think about perceptions within the within an environment, and we know that perceived social cohesion, perceived safety, and perceived social environment in general all relate to cardiovascular outcomes. And then finally, there's work to support this connection between crime as a social determinant and a social environment measure, and its relationship with changes in blood pressure in populations. We also know there's a direct association between psychosocial factors and incident cardiovascular disease. For instance, chronic stress has been related to incident cardiovascular death, particularly those with what are thought to be stable cardiovascular disease. adverse childhood experiences are related to the development of type 2 diabetes, and depression is a key psychosocial factor related to the development of cardiovascular disease. In addition, we know discrimination relates to cardiovascular events, as does social isolation, which has been affected really by the COVID-19 pandemic. And so to get into a little bit of how we understand how social determinants really get under the skin and lead to the development of cardiovascular disease, we're getting more and more insights into key direct pathways that particularly relate social determinants to immune response. For instance, we know that hematopoietic axis relates to how immune cells function. We know that sympathoadrenal and HPA axis, activation of those axes, can also affect the immune cells, as can glucocorticoid and catecholamine signaling that can be upregulated in the setting of chronic stress related to social determinants or adverse social conditions. But there are also, of course, indirect connections where these stressors lead to unhealthy behaviors that are directly correlated with chronic stress and ultimately lead to the development of cardiovascular disease risk factors and poor outcomes. So my lab in particular has been very interested in how these social determinants in particular lead to the development of obesity, and there are direct connections by which social determinants affect adipose tissue in particular. These stressors can again activate the stress-related pathways, but also affect the infiltration of more inflammatory immune cells into adipose tissue, which promote increasing release of inflammatory cytokines that promote insulin resistance, as well as more inflammatory, particularly visceral adiposity that leads to poor cardiovascular outcomes. And we also know that the release of pro-inflammatory adipokines, particularly from adipose tissue, also affects cardiovascular outcomes. And so if we're thinking about ways in which we can address these connections between social determinants and cardiometabolic health, we need to really think about designing multi-level interventions that not just only target the individual, but also work within the community and also address policies to improve access to resources for maintaining and improving cardiovascular health. And as was mentioned earlier, it's really important to really bring the community into the conversation, really center community voices in the development of these types of interventions. And so just to give you a sense of intervention we're working on and how it really tries to build on a multi-level framework, but also how it works to really center voices from the community. We've developed an intervention called Step It Up, which is an adaptive physical activity intervention. It's designed as a randomized trial to test whether or not using digital health technology that promotes physical activity can improve physical activity amongst communities in resource-limited areas of Washington, D.C. And this intervention was really built through an iterative process with community engagement. We've worked with a community advisory board called the DC Cardiovascular Health and Obesity Collaborative, or DC CHOC, through the entire process of developing and implementing this intervention. It's really looking at whether we can use more messaging around place that's really tailored to where people live to see if we can improve their physical activity. But we're building in those pieces to really look at how social determinants affect cardiovascular events and biology by really looking at how physical activity can mitigate some of these biomarkers related to social determinants. We've built this intervention in an iterative process where we've taken a phased approach to build up to our current randomized intervention. And then we also need to consider how these interventions can fit within a healthcare system as well, where we need to again bring community engagement even to interventions that are around clinical care, where we bring in team members who can affect social needs and who can create resources to link patients to social resources to address social needs within the healthcare setting. And so if we are really going to build successful multi-level interventions that are available, accessible, and affordable for populations who need help in improving cardiovascular health, again, it's critical to engage communities from the beginning. And with the efforts to build these types of multi-level interventions, we really have the opportunity to improve cardiovascular health, to better understand the biology of adversity, how social determinants get under the skin, and really promote more health equity in cardiovascular outcomes. So I'd like to acknowledge those who have done much of the work is related to the paper, but also to our Step It Up intervention. And I'm happy to take any questions. Thank you so much.
Thank you so much. I'm not sure why my video is not working. Thank you so much. I would like to ask anybody who has questions to put them in the Q&A. We have one right now. So what social determinants of health might be affecting South Asian Indian populations who have higher burden of adverse CVD outcomes despite having higher SES status in the US?
That's a great question. There's certainly work looking at factors such as acculturation that may play a role in cardiovascular outcomes related, particularly in South Asian populations. There are also factors related to discrimination and racism that affect populations across socioeconomic levels. And so I think one thing to really understand and keep in mind is that many of these factors are intersectional, but also that in many ways, despite level of resources, you can still be affected by social determinants of health. I think all populations in some way, in many ways, are affected by different social determinants, but and some are more likely to be affected by adverse social determinants. But we all have, despite economic resources or educational resources, we still am susceptible to the effects of adverse social conditions and stressors.
Thank you. One listener is asking, does training in cognitive behavioral stress management techniques have any role here?
I think that's a good point. I think there definitely is a role for thinking about how do you build stress management into some of the interventions that are more multi-level. I don't think individual level stress management in and of itself is necessarily the key to addressing social factors that are more structural, but I think combining, structural interventions with more with stress management tools that can really build resilience are some of the ways in which we can help communities most affected by adverse social conditions.
A third question, is there any data collected on the DC CHOC study on the impact of ambient noise, especially at night, such as ambulances, police, helicopters, on cardiovascular health?
So it's a good point. We aren't currently collecting that data, but if there was interest in working collaboratively to bring some of those tools to measure that into our study, I'd be happy to meet with someone. If they want to email me, I'd be happy to chat more about that.
Okay. An opportunity for collaboration.
Yes.
I don't know if any of the other panelists have any any questions or OBSSR staff. There aren't any other questions in the chat right now. Friday afternoon, I think maybe questions are slowing down maybe. But thank you so much for a wonderful and very comprehensive talk. And then we will move along to the final speaker in this session, who is Dr. Brenda Curtis, a principal investigator in the Technology and Translational Research Unit at the National Institute on Drug Abuse at the NIH. So Dr. Curtis, I will let you take it away. Thank you. You're muted. Uh-oh. I still can't hear you. Maybe our technical people can.
Your
There we-
headset? Is your headset?
Hello?
There we go.
You know how embarrassing it is that I'm like, I'm chief of the technology and translational research unit and I can't use my headset. Thank you, everyone. That is, hopefully the rest of the presentation goes better. Okay, let me share my screen. Okay, I will assume that if people can't see me or hear me, they will tell me. So, thank you for having me today. Today, I'd like to talk about the stressors that we encountered during COVID, including things like racial bias and coronavirus victimization distress, and how that relates to substance use. I have no financial disclosures or conflicts of interest with the presented materials in this presentation. And I'd like to start with giving a thanks to my collaborators. Fundamental to this research was Dr. Celia Fisher, who developed and validated two of the scales that we used in this study, specifically the coronavirus victimization distress scale and the coronavirus racial bias scale. She was also part of the thought plan and the data analysis for this study. And her PhD student, Zhang Yu, really worked, she did an internship in my lab and she conducted the analyses for the study and really helped us kind of, figure out what was going on in the population. And I'd also like to thank Sal and Ting Ting from my lab, who also helped with the initial design of the national study, the national longitudinal study, as well as implementation in all parts, including data analysis. So big thanks and shout out to them. Before the COVID-19 pandemic started, racial and ethnic minority adults reported less alcohol use and other drug use compared to their white counterparts. However, minority groups experience higher diagnostic rates of substance use disorder. So typically, they are diagnosed more with substance use disorders, and they receive inadequate substance use disorder treatment, as well as they experience more negative substance use related outcomes, such as being put in jail for drug-related offenses. Blacks, Asians, and Hispanics were disproportionately impacted by the COVID-19 pandemic and were more likely to experience coronavirus racial-related discrimination. In addition, there were preliminary indicators that racial and ethnic minorities were reporting larger increase in substance use during the pandemic than non-Hispanic whites. Also, there was a concern about pre-existing conditions, but more specifically, pre-existing racial inequalities in substance use diagnosis, treatment, and outcomes that highlighted the significance of research examining factors that could exacerbate substance use disparities during COVID-19 pandemic. Individuals' psychological distress has significantly increased during the pandemic. I mean, originating from worries about coronavirus infection, um, financial disruption, as well as victimizations that were experienced um, when there was like mistreatment due to coronavirus infection. And consistent with research on the coping role of drugs towards acute and chronic distress, there were studies that were reported during the early parts of COVID that individuals were tending to use substances to cope with the pandemic. So we were really concerned about this, and we were also concerned about distress relating to employment and housing disruptions during the pandemic, which we know from other types of, Katrina, Hurricane Sandy, other types of natural disasters, and this was a global disaster, but other types of natural disasters when when employment and housing disruptions happen, as well as stress relating to the episode. And we know that racial and ethnic minorities are disproportionately impacted by these types of situations. So, in addition to higher levels of coronavirus victimization distress, racial ethnic minority individuals may experience an increase in societal bias against their racial or ethnic group due to the pandemic. Most Asians experience racial bias motivated by racism, xenophobia, and the presumption origin of COVID 19. Blacks and Hispanic groups experience the increase in COVID related racial bias, typically corresponding by the disproportionately higher rates of COVID 19 infection. For Asian, Blacks, and other people of colors, including Hispanics, young adults, for example, had increased levels of coronavirus racial bias that was associated with depressive and anxiety symptoms. So for this study that I'm going to talk about today, we examine racial and ethnic differences, uh, biases, by examining the coronavirus victimization, distress, and coronavirus racial bias, as well as other stressors, such as housing and employment disruptions, looking at their association with substance use related risk. And in the sample, we will be talking about Asians, Blacks, Hispanics, as well as non Hispanic whites. And this is during the COVID pandemic. And when I talk about substance use disorders or substance use disorder risk, I am including all substances, including alcohol, tobacco, cannabis, and other drugs. We hypothesized that racial and ethnic minorities would report higher levels of coronavirus victimization, um, COVID racial bias, as well as other stressors, and that would relate to higher levels of substance use disorder risk. Okay, so let me tell you a little bit more about how we conducted the study and the study design. So this was also part of a larger longitudinal study. But for this analysis, we looked at the first 60 days of data. At baseline, we collected demographic information as well as some economic and housing information. Then we did a 30-day EMA, ecological momentary assessment. In this case, we didn't do it throughout the day. It was more like a daily diary, so an end of day questionnaire. And then 30 days after that, which was 60 days after baseline, we gave another survey to participants. And this one right here included, demographic information, housing disruptions, things like that, but also the coronavirus victimization distress scale and the coronavirus racial bias scale. Okay, a little bit more specific about the study design. As I mentioned earlier, this was a national sample. We had just over 1,300 adults. We collected the data not at the beginning of COVID, which I'm actually really glad we did because we got to get kind of like once COVID has kind of set up in the United States. Um, but we collected data from December 2020 to June 2021. The population was about 70% non-Hispanic white, 8.5% Asian, 11% Latino, and 0.5% African American or Black. Demographic information included self-report, race, ethnicity, gender, um, household income, education level, and we also collected 7 medical conditions that were identified by CDC as being associated with severe risk for COVID-19. So this included things like obesity, high blood pressure, diabetes, heart disease, cancer, and HIV. We looked at changes of employment due to pandemic and associated stress, as well as changes dealing with housing disruptions. The coronavirus victimization distress scale assessed 5 coronavirus victimization experiences and associated distress. Items include things like being teased or bullied, um, physically threatened, mistreated, verbally taunted and abused, being called bad names, as well as cyberbullying. The coronavirus racial bias scale is a 9-item scale, and here it looked at coronavirus being negatively affecting social attribute towards one's race or ethnicity. Things like I believe the country has become more dangerous for people in my racial ethnic group because of the fear of coronavirus, or things like there's negative social media posts against people of my race or ethnicity related to COVID. And also throughout, we collected data. We did the EMA data that asked about every day about substance use, but we also collected at baseline and at the 60-day. We did the NIDA modified assist, which looks at substance use. We did modify it to include like the last 30 days. So it would capture the 30 days, the time before the study, and then the 30 days after the EMA had ended. And we collected data on alcohol, smoking, and other substance use. So looking at some of the demographic variables of what was experienced relating to employment and housing disruption, about 66% of participants reported employment changes due to the pandemic, and about 52% reported that they were at least slightly troubled by these employment changes. Um, 25% of the participants reported housing disruptions due to the pandemic, which resulted in a significant age difference across race and ethnicity. Asians were slightly more likely to report at least one COVID-19 health risk than others, and both Asians and non-Hispanic white adults were less likely to report employment or housing disruptions due to the pandemic when compared to Black and Hispanic adults. Approximately half of the respondents endorsed at least one coronavirus racial bias belief statement. There were significant racial ethnic differences in coronavirus victimization distress, and racial bias beliefs were found with and using ANOVA as well as Bonferroni post hoc comparisons. Asians were significantly more likely to experience coronavirus victimization incidents than Hispanics, Blacks, or non-Hispanic whites. Moreover, most racial ethnic minority respondents reported at least one coronavirus racial belief, while 31 or 32% of non-Hispanic whites did. Asians reported the highest level of coronavirus racial bias among all groups, and this was significant. And Blacks and Hispanic participants also reported significant higher levels of coronavirus racial bias than non-Hispanic whites. More than half of the respondents were at moderate or at high risk of alcohol use disorder, and more than a third were at risk of tobacco use disorder. A quarter were at risk of cannabis use disorder, and about 20% were at risk for other drug use disorders. Among Blacks, about 35% reported tobacco use that would be classified as potentially having a tobacco use disorder, and about 31% for other drugs. And non-Hispanic whites reported about 28% for tobacco and about 24% for other drugs. And these were significantly higher risk of tobacco and other drugs when compared to Asians and Hispanics. Looking at the correlational relationship that's presented here, you can see that the coronavirus victimization distress scale and the racial bias scale were positively associated with each other. Risk for tobacco, cannabis, and other drug use disorders were positively associated with the coronavirus victimization scale, whereas alcohol use disorder risk was unrelated to the coronavirus victimization scale. None of the SUD risks were associated with the coronavirus racial bias scale. Considering the imbalance in sample size that I reported earlier, that was across the racial and ethnic groups, here we conducted a regression analysis was conducted with the same logistic regressions, but for each ethnic group. Coronavirus racial bias was associated with higher odds of alcohol use among Black adults. And for Hispanics, higher levels of coronavirus victimization distress were associated with higher odds of tobacco use risk. For non-Hispanic white adults, higher levels of coronavirus racial bias belief were associated with higher odds of tobacco use disorder, as well as other drug use risk. Sorry, not disorder, risk. Despite the higher levels of coronavirus victimization distress and racial bias among Asians, coronavirus victimization distress and coronavirus racial bias were unrelated to substance use disorder risk. So what are some of the takeaways of the study? Well, for one thing that we saw regarding stressors, Asians were significantly less likely to report were significantly more likely to report at least one COVID-19 health risk than others. Both Asians and non-Hispanic white adults were less likely to report employment and housing disruptions due to the pandemic compared to Blacks and Hispanics. However, there were no racial ethnic differences on employment disruption distress. Substance use risk was significantly increased in the US due to COVID-19, and we saw this in many other studies, and we also saw substance use risk being high in our population. The current study adds to a small but growing body of research on two of the coronavirus stressors, or at least actually more than two. We have the coronavirus victimization distress, we have racial bias, but we also have information around housing disruption and income. And so on coronavirus racial bias belief was associated with higher odds of alcohol use risk among Black adults. And we saw that coronavirus racial bias belief was associated with higher odds of tobacco and other drug use risk among non-Hispanic white adults. And for Hispanics, coronavirus victimization distress was associated with higher odds of tobacco use. These findings underscore the significance of examining how the current pandemic has exaggerated, racial and ethnic systemic inequalities, including the high rates of coronavirus victimization and racial bias beliefs that were experienced among racial and ethnic minorities. The study also calls for further empirical research on pandemic-related risk factors for substance use and substance use treatment sensitivity, being sensitive to specific needs of socially marginalized populations. I'd like to say thank you for, listening to my talk, and I would like to thank my current lab members. Without their support and their work, this research would not be possible. And I also like to give special thanks to Doctors Amy Newman, Nora Volkoff, and Lorenzo Leggio. They were all instrumental in providing support and resources so that we could conduct this national study and helping to get it going when NIH basically a lot of the research had closed during the beginning parts of COVID. And Dr. Leggio is also my branch chief and was instrumental in helping with the design and implementation of the study. And as much, my research is funded by NIDA and the NIDA Intramural Research Program. So thank you.
Thank you so much for a great talk. We have some questions in the chat. So I'm going to start with a question about your sample size. There's a question about why Black, Hispanic, and Asian subjects make up just about 25% of the study sample when these categories of populations got affected by the pandemic the most.
Yes, we did try to oversample. It was one of the, I mean, for an online study, it was actually considered, pretty high levels. But it was a national study, and we try to oversample for different populations. It was just basically who we got for the study. Yeah.
There's another question asking if you can talk about COVID racial bias in older adults in community living and those living in nursing homes.
For this study, we did not get people that were living in nursing homes. So I can't report from them for the data for this study, unfortunately. But what I will say is that we, in general, a lot of times don't do very well of getting older adults in studies. National Institute of Aging, there are researchers there who do a great job and who have been working with that. But for this study, we were not able to access the nursing homes. For one thing, we couldn't get into the nursing homes to do the research and for the panel companies to be able to recruit participants.
And I do know that NIA, the National Institute on Aging, has funded some research looking at community-based and nursing home dwelling older adults. I don't know if they look at racial bias, but I do know that there's something out there, so the listeners can do a little searching that way. There is a question. All right, hold on a second here. It wasn't. This person asks, I don't think you share the two scales. How is the coronavirus racial bias scale different from what would be considered typical, quote unquote, racial bias in healthcare?
Give me. I pulled it. Yeah, it's like 1 second. I had it up in the screen. Sure. It died. So the coronavirus racial bias scale, what was the question about the coronavirus racial bias?
So how does it differ from a quote unquote typical racial bias in healthcare?
Well, so it wasn't about healthcare. These were questions about, like, I believe the country has become more dangerous for people in my racial ethnic group because of fear of COVID. People in my racial ethnic group are more likely to lose their jobs because of COVID. I worry about people thinking I have coronavirus simply because of my race or ethnicity. So these were questions specifically, I mean, Dr. Fisher did an awesome job developing the scale and validating the scale, but these were specifically questions about how people of different racial groups were experiencing COVID in relation to what was being said in the media. When it first came out that certain groups were more affected than others, and then there were certain groups were being accused as being the carriers of COVID, and so it really tapped into what was going on at the time, or potentially still is going on.
Still going on. Thank you. No, that's really helpful. Thank you. Is there any comment? And there's like you said, there's been a lot of research on this. Any comment on how everybody started drinking more?
Well, we saw that people at least were having, we were pretty high. I think it was like 60-some% of people that were drinking at a level that if you were a healthcare provider, you should be doing screening and brief intervention. It was at that level of risk. So yes, there was, as you saw, it wasn't significant necessarily between the different groups, but everyone was drinking, or a lot people in the study were drinking at higher rates or at high rates. I don't want to say higher because I didn't do pre-COVID, so I don't
Right.
know.
There's other studies to indicate that that is the case, though, that
Right.
drinking across the board went up.
Yeah.
Did you see any factors that buffered against the experience of COVID-related victimizations, or would you hypothesize about any that there might be?
The question of resilience does come up a lot. And I'm going to say no, necessarily. I mean, we did see that older adults it seemed racial coronavirus, racial bias seem to be, if you want to use the word buffered. I don't necessarily know if that was it, or is it they have lived through so much racial bias, in their life that they were just like, yep, saw this one coming. So they, their
Yeah.
levels of reporting were a little lower.
Yeah.
But,
I do remember hearing anecdotes about certain groups saying, yeah, I'm not wearing a mask because we're already being profiled, going into stores or something with a mask is just going to make that worse, or that sort of thing. So I can imagine.
I mean, yeah, there were concerns. I mean, there were some states that had laws on the books that you couldn't wear masks, and people were concerned. But I will say that we did ask in the study about masking, and I've only looked at the data so far with African Americans and people of color, but they were supporting mask use, and they felt that it was protective for their population.
Great. I don't see any other questions in the chat, in the Q&A. If
Okay.
there are any, get them in there quickly. or we'll break a little bit earlier and take a break, and then come back here by 3:10. Thank you again for this great
Thank you for
session.
having me.
Absolutely. And this was a great session on social determinants of health. And we'll see everyone back here at 3:10. Thank you.
Hi, everyone. Welcome back. It is 3:10 here on the East Coast. So we are going to go ahead and kick off this last session of the 2022 Behavioral and Social Sciences Research Festival. It has been a wonderful 2-day event, and I am very excited to moderate this next session on measurement in behavioral and social sciences research. My name is Dr. Maureen Monahan Center. I'm a program director for diabetes behavioral science at NIDDK here at NIH. And for this last session, we will have 3 presenters. Each will give a 15-minute presentation with 10 minutes for questions after each presentation. Please, as a reminder, use the Q&A function to submit your questions. It has been great to see the engagement this afternoon, and I really encourage everyone to stick with us, and please add your questions for each of our wonderful presenters. We are going to start this session with Dr. Catherine Keyes. Dr. Keyes is a professor of epidemiology at the Columbia University Mailman School of Public Health, and she will be talking with us today about suicide in the agent-host environment triad, leveraging surveillance sources to inform prevention. Do you want to go ahead and start sharing your slides, Dr. Keyes?
Great, thanks so much.
All right, thank you.
Can you hear me?
Yes, I can hear you.
Oh, great. Let me just, can you see my slides or the presenter?
I see your slides. It's not presenter mode, so you're good.
Oh, great. Okay. Well, thank you so much for the opportunity to present here today on the work that my research group has been doing on suicide in the US and how it fits into classical concepts of the agent-host environment triad. I'm going to start with just a few basics on what we're talking about when we talk about suicide and suicide death. A suicide death is a death in which the descendant intended to die and implemented the means themselves. These designations of intentional death are often done by a medical examiner or a coroner, and usually need to have either a reasonable medical certainty when a medical examiner is designating the death, or so-called beyond the reasonable doubt. Because of this, the assessment of intent can be very challenging, and it remains controversial. And usually what it means is that we often underestimate the burden of suicide death within and across populations. This is especially true in drug-related deaths, where someone who has been suffering for addiction for a long time. It's not clear whether it was an intentional or an unintentional death, but designations of suicide death can be challenging no matter what the means of suicide death is. In the United
States. Interrupt you, Dr. Keyes. I'm so sorry. I'm not sure if it's just me, but on my screen, the very right edge of your slide is somewhat cutting off. So I don't know if others see that or if there's a way to change the orientation.
Same thing for me.
Just wanted to give you a heads up, so I'm sure there's very important information on the full slides.
Yeah, I don't know. It looks okay on my screen. So if there's anything that's cut off, I can share the slide after the end of this presentation. So hopefully, is it fully cut off or just a little bit cut off?
Just a little bit.
Okay, I think it should be okay moving forward, but I can also share the slides if there's anything people are missing. So when we look at trends over time in recorded suicide death in the United States, we can take a very long view. These are data on the recorded suicide deaths in the US from 1900 through 2020. And you can see that compared to the early parts of the 20th century, suicide death is substantially lower in recent decades. But what's also really clear from these data is that if you compare the suicide death rate in the United States in 2020 to the recorded suicide death rate in 1990, it really has been unchanged in the past 100 years, over 100 years, even though there have been increases and decreases. And what's particularly relevant, I think, for our discussions now is that we had a period of real declines in suicide death in the later part of the 20th century. And that really reversed just around the year 2000. And since around the year 2000, suicide deaths have been increasing approximately annually in the United States. And in total, suicide deaths have increased about 30% in the last 20 years. These increases in suicide deaths have not been randomly distributed in the population. And what I'll talk about today is how they correlate both spatially and temporally across the United States. What you see here are the death rates from suicide in the US, stratified by sex for men and women, and by firearm death, which is a predominant means of suicide death in the United States. Each one of these hexagons in this picture represent a suicide death rate arranged by age on this axis, period on the top axis, and cohort on the left axis. And we could spend our entire time just dissecting these patterns in this picture, but a few things that I want to point out to us. One is that we have a real concentration of high death rates from firearm suicides in the US for older adult men. Suicide death rates really accelerate from firearms in the United States among men after about age 65 and 70. Also, you can see this clustering of increasingly red hexagons through the life course in the later periods of this data series. And what this indicates is a concentration of increasing suicide deaths among men in midlife in recent years. We see similar increases among men in midlife, slightly younger, through non-firearm means of suicide death. So it's really this concentration of firearm suicide deaths among men in this middle period of life. For women, suicide death rates are much lower than they are for men, but we can still see this increase in the suicide death rates among later periods, among women earlier in the life course, in the 40 and 50 range. And the other thing to point out is that you can kind of see this increasing acceleration of suicide deaths among both men and women at younger points in the life course, individuals in young adulthood as well. Another aspect of suicide epidemiology that I think it's really important to point out is that suicide deaths are relatively rare in the population, whereas suicidal thoughts and even suicide attempts are relatively common. These are data from the Youth Risk Behavior Surveillance Survey, showing trends over time among high school students in suicidal ideation, making a plan for suicide, attempting suicide, and attempting suicide in a way that was injurious enough to seek medical attention. And I just want to point out how common these experiences are for adolescents. You know, by about 2019, about 1 in 5 adolescents had suicidal ideation in the past year, and just under 10% attempted suicide in the last year, a non-fatal suicide attempt. We know that the epidemiology of suicidal ideation and even non-fatal suicide attempts is quite different than death by suicide. For example, men are more likely to die by suicide, whereas women are more likely to have suicidal ideation and suicide attempts. What we do know is from social autopsy studies is that approximately 90% of people who die by suicide have a preceding psychiatric disorder, most often mood disorders like depression and other depressive disorders. But most people who experience these common mental health problems will not go on to die by suicide. And in fact, what we know is that a suicidal crisis is often, but not always, quite short in duration. And so that window between deciding to attempt suicide and a suicide death is very short. It's often less than 1 hour and really requires a crisis response. When we are able to disrupt that process and stop a suicide attempt during that suicidal crisis, it's life-saving. We know that approximately 90% of people who survive a suicide attempt do not go on to reattempt. And what this really indicates is that during that suicidal crisis, reducing the access to lethal means, for example, a firearm, is a really key factor in determining fatal versus a non-fatal suicidal act. And going back to firearms, we know that in the United States, firearms are the predominant cause of suicide death. They are a very lethal means of a suicide attempt. It's very difficult to recover from a self-inflicted gunshot wound. Currently, about 53% of suicide deaths are by firearm in the United States, and asphyxiation, including suffocation, remains the second most common. Another piece of this epidemiology that I'd like to point out is that there are some changes over time in the proportion of, among people who die by suicide, the proportion that are using different means. This is a study that my research group just published this year, showing time trends among adolescents aged 10 to 19 who died by suicide and what means they used. And something important to point out is that deaths from asphyxiation, especially among female adolescents, are rapidly increasing in the United States as the means of suicide death. And that poses a real challenge for public health. We have a pretty robust set of prevention tools that we have developed in order to keep people safe who have, for example, a firearm in the home, which increases the risk of a death by suicide by firearm. And asphyxiation deaths, including suffocation, are much harder to restrict access to in non-institutionalized settings, and really requires us, in terms of a public health response, to really focus on those root causes of what is precipitating a suicidal crisis, and focusing on the prevention and intervention of the psychiatric disorders that so often precede a suicidal act. So thinking through this epidemiology, my research group has really focused on positioning suicide in terms of its spatial and temporal correlations within a classic infectious disease model, thinking about host, agent, and environmental factors. Host factors are often those that we think about a lot when we think about suicide risk, like having a psychiatric disorder history or previously attempting suicide. But what my research group has really focused on is these agent and environmental factors. Agent factors being things like owning a firearm or having a firearm in the home, as well as factors like media reporting, which I'll mention, and environmental factors as well, such as seasonality, temperature, and altitude. Thinking through these kind of short-term temporal correlations and media reporting, we've known for hundreds of years that there can be increases in suicide death with inappropriate reporting of suicide deaths, as well as fictionalized accounts. This is the book, The Sorrows of Young Werther, published in 1774, in which the main character has unrequited love and decides to end his own life. This book was very compelling, and at the time there became an increase in the suicide death rates among men who were similar ages and who even dressed in the same style as the character in the book. So much so that the book itself was banned in several countries shortly after its publication. We know from numerous studies that media reporting of high-profile suicide deaths increased the population rate of suicide across many countries and across many time periods. This is a meta-analysis that was just published several years ago, updating what we know about the impact of inappropriate media reporting of suicide deaths. And the take-home message is that on average, in the 30 to 60 days after a inappropriate reporting of a celebrity suicide, population rates of suicide increase about 13%. Here in the United States, the most recent example of this was the death of the entertainer Robin Williams that occurred in 2014. And shown here are the monthly number of suicides in the United States for the years preceding the death of Robin Williams. And this red line here was what occurred in the United States very quickly after Williams' death. We saw a tremendous increase in the number of suicides in the US. And using spatial temporal models, accounting for seasonality and long-term trends, we estimated that there were over 1,800 suicides in the United States in the 6 months following Robin Williams' death that was attributed to the inappropriate media reporting. This is positioned alongside a long literature, including work done by my colleague Maddie Gould at Columbia and others, about suicide clusters, really how suicide events can be transmitted from person to person, through these different point source outbreaks, as well as more traditional person-to-person transmission. And we've seen increasing evidence of this in the United States as well. Shown here is a paper that we published this year, identifying spatial and temporal clusters of suicide in the United States among young people, people aged 5 to 25 in the United States. And I'd just like to orient you to what I'm showing you here, focusing here on all suicides. This x-axis is time going from 2000 to 2020. Each of these lines represents a cluster of suicides. And by that, we mean an area of the United States where the suicide rate was higher than what we would anticipate if suicides were randomly distributed across the US. These numbers here represent the number of counties in the United States in each of these clusters. So for example, right here, in right about 2005, 2006, there was a cluster of 11 counties in the United States where the suicide rate was 4 times higher living in that 11 county geographic region than we would have anticipated if suicide rates were randomly distributed in the United States. And just a few things I'd like to point out is that you can see across time just this increasing number of suicide clusters. Both the size and the number of clusters is really increasing, indicating that there's just increasing spatial aggregation of those suicide deaths. You can see down here these red clusters. These are what we call cold clusters, where the suicide rate is lower than we would anticipate if suicides were randomly distributed in the United States. And those are increasing as well, which indicates that there's just increasing heterogeneity in the United States between areas with high suicide rates and areas with low suicide rates. And there's also racial ethnic disparities in where suicide rates are occurring and to whom they are affecting. There's a whole lot of other research on other spatial elements of suicide risk. We know, for example, that ambient temperature, sunlight duration, both in the United States and elsewhere, influence the suicide risk, as does altitude. Areas with high altitude in the United States have higher rates of suicide. I'd like to end the presentation talking a little bit about COVID-19 and what's been going on with suicide during COVID-19 and our own research where we're trying to leverage novel surveillance sources. There was a lot of concern, especially in the initial periods of the pandemic, about a potential increase in suicide rates in the United States. We saw in these reviews and meta-analyses confirm that there was an increase in population level rates of psychological distress, as well as psychiatric disorders among both adolescents and adults who were enduring this ongoing pandemic that we've all been facing. And we know that psychological distress and psychiatric disorders are among the strongest risk factors for suicide. And so there was a lot of concern among all of us that we're going to see a population level increase in the suicide rate. Fortunately, what we've learned is that in many countries, including the United States, there hasn't been the anticipated increase in the suicide rate during the COVID-19 pandemic that we would have expected, given the increase in these risk factors. This is a paper of 33 different countries using interrupted time series to estimate changes in the suicide rates across all of these different countries. And these countries up here in green are countries where there's very good evidence that the suicide rate didn't increase and in fact declined in many countries during the pandemic, including the United States. these countries down here are countries where there's strong evidence that the suicide rate did increase. So one take home is that there is a lot of heterogeneity in terms of COVID-19 effects. In Japan, for example, there's good evidence that suicide rates have increased, but they haven't in the United States, contrary to what we would have expected. In my research group, early on in the pandemic, one problem we have is that there's a really long lag time before we get finalized, confirmed death data for outcomes like suicide. They require investigation, they require these designations of intent, and we don't want to wait for that in terms of disseminating a public health response to crises like COVID-19 and potential mental health problems. So early on in the pandemic, along with my researchers in infectious disease who use novel surveillance sources to track things like flu and COVID, we used Google data to track trends over time in searches for suicide-related search terms that are relatively well validated in various literatures. And what I'm going to show you first here is our Google data. We use Google data we use cell phone tracking data to understand the proportion of people in the United States who left their cell phone completely at home for a 24-hour period. And we correlated that with all kinds of Google data on outcomes. So shown here was kind of our proof of concept. We looked at economic stressors, which we know to be causally related to COVID-related shutdowns and COVID infection rates across the US. Indeed, these are Google searches for economic stressors like being laid off, collecting unemployment benefits, looking for a job, things like that. And what I'm showing you down here is the cross-correlation function for the association of those Google search terms with the proportion of time that devices were left completely at home. And shown here in red is a significant correlation between the proportion of devices that were left completely at home and how many people were searching for economic related search terms. So we thought this was a good proof of concept. We know there's a causal connection between these two things, and we can detect it in our data. We then went on and looked at suicide related search terms. And I'm showing you here just one example from search terms related to seeking information about how to die by suicide on the internet. And we didn't see the same big spikes in the initial phases of the pandemic in these related search terms. And as you can see down here, we also didn't see really, you don't really see any of these red squares, which are indicative of this positive correlation that we expected to see. We really didn't see any evidence that there was an association between the time devices were left completely at home and these suicide-related search terms. And when we published the paper, we thought, well, maybe Google data are just messy. We don't really have any information on the true intent of why people are Googling certain search terms. So maybe Google data just aren't a strong enough signal for us to detect this. But once the data came out showing that there really was no increase in suicide death rates in the United States, we realized that the Google data perhaps, we don't know, but we thought the Google data might actually be a good sentinel indicator of what's going on underlying the population in terms of suicide-related risk. And the Google data might be an appropriate way forward to start to detect anomalous increases that we could use to disseminate public health interventions. So I would just like to close there. I know I mentioned that The Sorrows of Young Werther was this book published in 1774, and it elicited this real increase in population rates of suicide. But there's also another common fictional work that people cite in this work, and that's the Papageno effect. Papageno was a character in The Magic Flute who, much like Werther, thought he was experiencing unrequited love and decided to end his own life. What's different in The Magic Flute is that the community came out. The community came forward and they told Papageno that his life was worth living and that he was a valuable member of the community. And Papageno didn't die by suicide because the community was there to support him. So I'd like to end with that, just noting that it's all of our responsibility to be good stewards of mental health in our communities and to disseminate important public health information related to mental health and suicide risk. And I would just like to close with thanking my funders, including NIMH, and my co-authors. Thank you very much.
Thank you so much. That was really a wonderful and informative presentation. We now do have a few minutes for question and answer. I do see one question that just thanks you for the wealth of information and asks about the availability of slides. We can certainly discuss that offline. But
I'm happy to send the slides.
Thank you. I do see another question. Could you please comment on the influence of internet and violent video games and movies on adolescents and young adults and their suicidal or violent behaviors?
Sure. I mean, on one hand, so there's been a lot of research into violent video games, for example, and associations with child mental health in a number of different domains. Typically, that research has focused on aggression and externalizing disorders as potential outcomes of video game use. And really what the research shows is it's quite heterogeneous. You know, there certainly is some small effect in terms of kids who typically have underlying vulnerabilities that might be exacerbated with repeated exposure to violent images, including video games, but also internet use. And that is true for other studies of digital technology and its association with child mental health. You know, certainly there is a signal there for some children, but you know, it's not across the board, you know, a detrimental outcome at a population level. The one thing I will say that people in the suicide field are increasingly concerned about is the availability on the internet for young people to learn about lethal means of suicide. And this is increasingly a concern in terms of how to protect kids online. And there's a lot of efforts, both in the United States and in other countries, to restrict access to these types of sites and to provide adolescents with more safe information about mental health and about suicide risk. But you know, we're butting up against a lot of other competing sources of information, and it's really troubling. And I do think the increase that we're seeing in asphyxiation deaths in the United States, this is just my opinion, I don't have data to back it up, but I do think that that's partly driven by information that young people find online. So that is very concerning to me.
Okay, great. Thank you so much. In our final minute, we do have one additional question. That's a very interesting presentation. Thank you. Given the lack of demonstrated association between COVID and increased rates of death by suicide, do you have thoughts about how to explain the increasing rates of suicide among some ethnic minority youth populations, such as Black, Native American, Alaska Native youth?
Yes, that is an area that we're increasingly focused on. I do find it very troubling. A lot of the concentrated increases we're seeing, especially among young people, are among adolescents who are racialized as minority in the US, including Black adolescents, but also Asian and Pacific Islander adolescents. I don't think we have good information at this point to know what is driving those particular increases. But I know that my research group and a lot of others are really working on this, not only to understand why it's occurring, but to develop tailored interventions that really speak to the concerns of youth in these communities. So I know that's not a great answer, but it is one that a lot of us are really focused on working on in the coming years.
Excellent. Well, we will look forward to seeing more of that data. And thank you again for really talking about these innovative surveillance strategies, and in particular, the implications potentially for clinicians or just-in-time interventions. Great to hear about this work. So thank you very much, Dr. Keys. We will now move to our second presentation of this session. And we have Dr. Edwin Aroke, who is an associate professor and director of the PhD program at the University of Alabama at Birmingham School of Nursing. And we will hear from him today about the congruence of neighborhood disadvantage and individual objective socioeconomic status as predictors of chronic low back pain. And I see your slides. Thank you so much.
All right. Thank you, Maureen. And thank you for having me. Again, I'm Edwin Aroke. We're going to share some of the work that we are doing in my lab. Can you hear me okay?
Yes, I can hear you fine. Thank you.
All right, that's good. All right, so moving right along. This will happen. All right. I have no conflicts of interest, but I would like to start by acknowledging the funders, which have really been grateful for all this work. It would not be possible without, the support of the NIH. I have an R01 from the National Institute of Arthritis, Musculoskeletal, and Skin Diseases. Also, part of this work was funded from an R01 that we have from the National Institute on Minority Health. And of course, I also have some intramural funding from UAB, which really helped lay the foundation. And most importantly, I really want to acknowledge some of the collaborators that we have in my lab. my biggest mentor, Dr. Dora Bowden, Dr. Telesbo, Crossman, they have really been helpful. The graduate students in our lab who collect the samples, and of course our research participants. All of this again would not be possible without their help. And just to give a little context, chronic low back pain is the leading cause of disabilities worldwide. And when you look at, most of us going through life, we take for granted the fact that, you can bend down and tie your shoes without really any problem, right? We take for granted the fact that you can walk upstairs and do anything that you want. The truth is, as we go through life, about 75 to 80% of us are going to experience back pain at least once in our lifetime. The good news is for most of us, it's going to go away, right? Maybe you stretch, maybe you exercise, maybe you take some Motrin, it's going to go away. Well, for some individuals, that back pain does last quite a bit longer than we really expect. In fact, after 1 year, about 60% of adults still do report back pain. So if the back pain lasts between 0 to 6 weeks, we call it acute low back pain. Somewhere between 6 to 12 weeks, we call it subacute low back pain. And beyond 12 weeks, we call it chronic low back pain. And again, when you look at most chronic conditions, this is one of the most disabling chronic conditions out there. And for adults who have chronic low back pain, about 85% of them have what we describe as non-specific chronic low back pain. Non-specific chronic low back pain basically means that if we were to do a CT scan or an MRI, we cannot identify any pathological cause. So we do not know why these individuals have back pain in the first place. And so one of the key questions that we've really strived to answer in my lab is, why is it that some adults do have non-specific chronic low back pain? Why is it that after a stretch or after taking Motrin, yoga, the back pain doesn't go away? And this is even more significant because, again, when you look at in terms of cost, one, low back pain is very costly. Pain overall is one of the most costly chronic diseases that we have out there. Then in terms of cost to the individual, it affects virtually every aspect of the individual's life. And pain, yes, affects individuals of all backgrounds, including individuals of various races, individuals of higher and lower socioeconomic status. But we know that individuals of lower socioeconomic status generally have more severe and disabling chronic pain, especially chronic low back pain, when we compare them to those from higher socioeconomic status. Again, of course, most of the time when we think of socioeconomic status, we usually would consider individual level factors. We think about things like individuals' income and education level, which have predictably really shown that lower levels of income or lower household income and low levels of education correlate strongly with more severe and disabling pain. Similarly, when you look at subjective socioeconomic status, multiple studies have correlated those two with chronic pain conditions. But as we look at environmental factors, we also know that, well, somebody's environment does play a role, right? So we've also tried to say, well, why some studies have really focused on the roles of the individual, right? As we really as we appreciate more about disparities, we realize that the environment does play a big role. And we know that living in a disadvantaged neighborhood has been associated with poorer chronic pain outcomes. So what we wanted to really do is understand what is the interplay between individual level factors and environmental level factors, specifically trying to understand that, well, when you look at individual level factors, it looks like the individual socioeconomic status may be mediated by factors such as the income, lower levels of education, and the availability of social resources. And those generally would be what predict the relationship with low back pain. We also know that at the community level, again, neighborhood deprivation does predict chronic low back pain. So the objective of this work that I'm going to be sharing with you was really to compare neighborhood deprivation and individual levels socioeconomic status to see which is a better predictor of chronic low back pain. So that if we were to do an intervention, should we be doing interventions at individual levels or at the community levels or both? Or do both really equally predict chronic low back pain? In terms of our measures, we measured pain severity and pain interference using the brief pain inventory. And in terms of individual level socioeconomic status, we measured the subjective socioeconomic status using MacArthur's ladder. And in this, we basically asked research participants to say, where do you see yourself on the social ladder compared to everybody else in the United States? Such that a score of 10 means that I am pretty well off compared to everybody else. And a score of 0 or 1 shows that, well, I think I'm really down there in the run of the ladder. So most people are better off than me. And this is really trying to say, how does the person see themselves? But in terms of the objective view, we also ask participants to tell us about their income, household income after taxes, and their level of education. Then to look at the community level, socioeconomic status, we use the residential area deprivation index. So the area deprivation index is basically a geographic area-based measure of disadvantaged positioning relative to society. And it's a composite score. And we calculate the ADI for the state of Alabama using a measure developed by Singh and al., which really uses the 17 US Census measures, which have all previously been associated with mortality. And as you see here, the measures include things like median family income, income disparities, the percent of owners occupied housing units in the area. Things also include things like percent of single parent household with children less than 18, and the percent of occupied housing unit. And basically, we use this to predict, again, neighborhood deprivation. Because previous research has suggested that neighborhood effects are non-linear, ADI scores were grouped into quantiles with the lowest quantile associated with lowest deprivation and higher socioeconomic status. So basically, the lower the score basically means they have higher socioeconomic status because there's less deprivation in those neighborhoods. So to basically compare this, we recruited about 104 participants in this pilot work, basically looking at, again, the difference between individual level and community level socioeconomic status. The average age was about 45. Most of our participants were female, 58%. A slight majority of them were African American, and we had about 41% white. This image shows you the simple linear regression, basically predicting, again, pain severity and pain interference, and using ADI, income, education level, and subjective socioeconomic status. In our sample, pain severity and pain interference correlated positively with ADI, but negatively with income education. And again, you can see here that the income does account for most of the variance compared to, or higher percentage of the variance compared to ADI, education, and subjective socioeconomic status. And this shows you a comparison of the different models that we use. And as you can see, income has the smallest AIC and BIC, basically saying that income performs better in predicting both pain severity and pain interference compared to ADI and education level. We've now also used regression models to try to predict, again, pain severity and pain interference. And what you see from our multiple regression models is that ADI makes a small independent contribution to both pain severity and pain interference. Income makes the largest contribution, but education makes a contribution to pain interference, but not pain severity. Which, if you think about it, especially when you think about education level, it makes so much sense. the level of education may be linked to the type of job that the research participants hold, such that those with lower levels of education may have more physically demanding job. Well, if you have a job that is very, very physically demanding, it means that, yes, for the same level of pain severity, the level of pain interference in your daily activities may be higher depending on your level of education and your associated job. So in terms of implications, we have previously also looked at the relationship between neighborhood deprivation and systemic inflammation in individuals with chronic low back pain. And we found that, again, in non-specific chronic low back pain, there is a correlation between neighborhood deprivation and serum levels of pro-inflammatory markers, specifically looking at CRP. We also looked at whether there was a relationship between neighborhood deprivation and sleep-related disorders, and found that, yes, individuals living in low socioeconomic status neighborhood tend to have higher levels of poor sleep, or chronic pain-related sleep disorders. And these findings have some important implications. One, this is really telling us that there is an independent contribution of neighborhood effect on chronic pain severity and interference. So neighborhood factors do predict pain severity, do predict pain interference, and that is not linked only to somebody's income or socioeconomic status. So there is something about the accessibility of the neighborhood, the greenness of the neighborhood, that may be affecting pain severity and pain interference. Also, it also means that neighborhood factors warrant further consideration, especially as we attempt to address social determinants of health. It means we cannot focus so much on individual factors and ignore neighborhood factors. We have to also be looking at policies that affect neighborhood factors, and also set policies to address neighborhood factors, not just individual level factors, if we are going to achieve health equity. Just to wrap up, I'm going to share with you, this just shows you some of the work that we are currently doing in this space. We have work looking at epigenetic changes, comparing looking at the role of epigenetic changes in racial disparities in pain, really trying to map out whether do epigenetic changes lead to gene expression changes and correlate with racial disparities in pain. We also are looking at the role of the epigenome and epigenomic changes in socioeconomic status disparities. Again, trying to compare both individual level factors and environmental level factors, in terms of epigenetic changes. Neighborhood deprivation, epigenetics, and accelerated aging. I have a PhD student who is really spearheading this work, trying to really understand the interplay between living in a poor neighborhood and age-related chronic conditions. And she hypothesizes that accelerated epigenetic aging is really what is driving the disparities that we see. Then we also have a little bit of work looking at the effect of heat islands and the greenness of various environments in their relation to pain outcomes. And these are some references. Thank you.
Thank you, Dr. Oroki, so much for that really excellent presentation. I really appreciate the multiple ways that you approached assessment for this work. And I think it really echoed a lot of the themes that we heard earlier today in the keynote address from the director of the National Institute of Environmental Health Sciences, really thinking about these broader neighborhood factors and epigenetics. This talk is open for questions, so please feel free to add questions to the Q&A box, or if you're currently a panelist and you have a question, you can just raise your hand or put it in the chat. I'm going to start with a question, if that's okay. Again, I really appreciated the multiple ways that you looked at assessment, and was interested in the inclusion, not just of the area deprivation index, but also looking at both objective and subjective social status using that MacArthur ladder. Can you talk a little bit more about the importance of incorporating both that objective and subjective social status in your work and how you've incorporated this into your research?
Sure. that is a very excellent question. So one thing that we have observed, even in our sample, is that just how much somebody makes doesn't necessarily reflect how they see themselves. And yes, we do look at objective measures such as household income, which is very informative. But even more important is how does the person see themselves? And this was really reflective in some of the work, which I didn't share that here, which I have previously published, looking at the relationship between subjective status and pain outcomes, and also looking at the interplay between that and racial disparities. So previously, what we found in that area was that when you look at disparities between Blacks and whites in terms of non-specific chronic low back pain, subjective social status, that relationship between subjective social status and pain interference is mediated by depressive symptoms, and also changes as a factor of somebody's race, such that for white individuals, the higher their subjective social status, the less depressive symptoms they reported and less pain interference. But we saw that where for Black individuals in the United States, higher subjective social status instead correlated with more depressive symptoms and more pain interference. So that really caught our attention to say, well, it means that when you are looking at the relationship between subjective social status and objective social status, it's not the same for everybody. And we did see this difference, especially when you start looking at how do Blacks see themselves and how do whites see themselves. And that has come to be important to us because, again, I think the way the person sees themselves is just as important as some of the objective measures are, if not more important.
Interesting. No, thank you. That is really great to hear your perspective on that and how you've incorporated into prior work, and also how it might be different for different people. But we do have a question that has come in asking about the area deprivation index. Have you examined if higher ADI correlates with other neighborhood level factors, maybe that weren't captured in the list, such as presence of food deserts, absence of green spaces, or walking paths? And what needs to be measured to help guide the efforts to eliminate some of those disparities?
Yes, so yes, higher ADIs definitely do correspond with some of these environmental deprivation factors. I think one area that has to be measured is really going beyond saying, well, let's use census data to predict where your zip code is and saying where do you sleep. I think other factors that have to be captured is walkability of the neighborhood, right? Those factors are important. For instance, if we are saying somebody has back pain and we say go out and exercise and walk, well, if you are living in a neighborhood where there are no sidewalks or it's not safe to go out and walk, then that intervention will not really work. Okay, so looking at greenness of the neighborhood, again, I did mention a little bit about the heat islands. And the whole concept basically being that if you look at generally poorer neighborhoods tend to be located in these heat islands. And those neighborhoods tend to have higher levels or higher prevalence of most chronic conditions, including chronic pain. So is there a relationship between the greenness of the neighborhood, for instance, and most of the chronic conditions that we see that disproportionately affect individuals of lower socioeconomic status? So being able to measure some of these other salient factors will be important, not just for scientific rigor, but also for policy. Right? We can easily make more neighborhoods more walkable. We can make sure that we make the neighborhood parks more accessible so that children can use the parks. Right? We can make sure that the parks are safe for everybody, that there are sidewalks for people to walk and ride their bikes and things like that. So that as providers, when we say go outside and walk more and do more exercise, it's safe to do so.
Yeah, no, that's great. And I definitely appreciate, thinking about moving beyond zip code. And the last part of this question was, did you have specific measures, in thinking about some of these things that you're looking at, like walkability, neighborhood safety, or absence of green space, other measures that you find to be helpful for these constructs?
We haven't specifically looked at any other measures, but what we've tried to look at is how long has somebody stayed in the neighborhood? It's a very intriguing question that we have been asking related to some of these factors to say, well, because again, for a cross-sectional study, we are measuring at a point in time, but how much of days really have stayed? And linking back to one of my main focus on epigenetics is saying, well, is it a factor of your neighborhood that is causing changes in the body, right? Because trying to look at how does the neighborhood get under the skin, as opposed to saying, well, there's just a correlation. And so trying to figure out at what point in a lifetime does some of this get under your skin. So I think it would be interesting to see what other measures others use. I know one of my doctoral students has been trying to use geo-specific data and some of the Google Maps to, map some of from some of the various neighborhoods to correlate them with various outcomes. So, but I think it's an area that warrants more consideration and more investigation.
Okay, great. Thank you so much. And in our final 2 minutes, I just have one final question for you. Again, really appreciate this kind of multi-component, multi-level look at chronic pain and low back pain. What do you see as the implications of this work for interventions that can improve health outcomes and well-being in this population?
I think it shows us that humans generally, or individuals are more than just a medical thing. That is, the biomedical view is very limited. When somebody comes in, and I speak from this perspective of a nurse anesthetic, it's upon me to look beyond saying this is just pain and back pain. Where does this person come from is going to be very important as we start thinking about interventions. Is it safe for this person to be able to live where they are? Do they have good sleep, right? So again, when I start looking at how the studies are all fitting together, we are seeing that, well, there's correlation with chronic inflammation. So yes, there's a biomedical aspect of that. How can we reduce that systemic inflammation? We are saying, well, it's correlating with poor sleep and sleep-related disorders. Well, if you're living in a noisy neighborhood, chances are you may not have good sleep. So could we work with policymakers in terms of those interventions and public health services to make sure that, hey, neighborhoods are respected? Also, like I said, for me, somebody who does translational work, I have been looking at the epigenetic part of it. One of the beauties of epigenetics is that it's reversible. So can we develop targeted interventions to reverse some of these epigenetic changes that we see in individuals with lower socioeconomic status compared to those with high socioeconomic status? So I think it speaks to really the fact that interventions have to happen at the individual level and at the community level if we are going to be successful, again, in bridging the gaps and the disparities that we see between low and high socioeconomic status as it relates to non-specific chronic low back.
Excellent. Thank you so much. And thank you again for a really outstanding and an informative presentation. We really appreciate it. And now we'll be moving on to our last presenter for this session. I'm pleased to introduce Dr. Nancy Kanwisher, who is an investigator at the McGovern Institute for Brain Research and the Walter A. Rosenbluth Professor of Cognitive Neuroscience in the Department of Brain and Cognitive Sciences at Massachusetts Institute of Technology. And she will be presenting today on category selective regions of the ventral pathway are present within a few months of birth. And so thank you so much. I see your slides and go ahead and take it away.
Great, thanks so much. Well, thanks so much for inviting me. It's an honor to be participating in this symposium. The starting point for the work that I want to talk about today is what I think is a pretty lovely picture of the functional organization of the cortex in adults, shown here, that our field has kind of worked out over the last few decades. So this picture is highly schematic. So what do I mean by sticking some colored blobs on the brain with some word labels? Well, what I mean is that each of these regions shown here has a quite distinctive, often very specific function. And we can see that by considering functional MRI responses to some of these regions. So this little blue region there is a region that I named the fusiform face area a very long time ago. And I named it that because here's what it does with functional MRI. This blue bar is the strength of the response of that region in functional MRI, which is indirectly but very correlated with neural activity in that region. When people are looking at pictures of faces, that's in blue. When they're looking at pictures of bodies, that's in purple. Places, that's green. Objects and words and other things, And so the point of this is that region really responds quite selectively to images of faces. Of course, that's not enough. It's also important to note that this region is a typical site for prosopagnosia, that is a selective loss of face recognition abilities after brain damage. And other causal studies have shown that, for example, electrically stimulating that region, which happens rarely but occasionally for clinical reasons in neuropsychology patients, neurosurgery patients, produces a percept of a face. Okay, but of course, that's not the only region here. This green region here is the parahippocampal place area, because here's what it does with functional MRI. It responds strongly when you look at images of places. Doesn't matter if they're indoor or outdoor or familiar or unfamiliar. Anytime you're looking at a scene, you get a strong response there, and much lower responses for bodies and objects and words. And this is faces right there, 0. 0 in these studies is the same as staring at a tiny dot on the screen. This purple region over here is the extrastriate body area. It responds more strongly when you look at images of bodies and body parts like hands and feet and knees. That's the purple bar, compared to faces and scenes and other things. And over here in kind of orangey yellow, that's a region known as the visual word form area, because it responds quite selectively to images of visually presented words and letter strings. That's this bar right here, compared to all of these other conditions. So those are a few examples of some of the results from functional MRI and other methods that have led to this kind of picture of selective regions of the brain. And so the diagram is meant to show that a set of regions that are present in more or less the same location in pretty much everyone. And I think this is exciting progress, because I like to think of this as a kind of initial rough sketch of who we are, what machines are running in our heads that make us who we are. So I think that's great. But this diagram also raises a huge number of questions. And the one I'm going to tackle today is a really obvious one, just looking at this diagram. And that is, how does all this structure get wired up in development? How do you start with 30,000 genes and a little bit of experience and get an organization a lot like this in pretty much every normal person? And so in this talk, I will focus mostly on this, what's called the ventral visual pathway involved in high-level visual perception here. This is the bottom view of the bottom surface of the brain, looking up from the bottom. And I'll consider in particular 3 questions. How early in development do these regions arise? What is the role of experience in the development of these regions? And I'll talk briefly about how each of these regions knows how to land in that particular part of the cortex. Okay, so as background, there are a number of studies that have been done in the past that have considered these questions. And many of these studies have argued that this face region right here develops very slowly and is in fact still developing straight through adolescence. And in fact, there's another study in monkeys. Monkeys have these many of these same regions, including the face and place and body selective regions. And one study showed that monkeys that were raised with normal visual experience, except that they never saw faces, that those monkeys don't develop face selective regions as normal monkeys do. And so these considerations led to the idea that the face area, at least, takes many years to develop and requires visual experience. And so those are interesting claims, but I would ask, really? Before we make these big conclusions, let's consider some other sources of data. So most of the prior neuroimaging studies before my collaborators started working on this were done on kids age 4 and up. And that's hard enough, but it's also too old. Like 4-year-olds are pretty developed. They know a lot of stuff. They've had lots of experience. If we really want to know basic questions about how things develop and what's built in, 4 feels a bit old. So what about infants? Really what we'd like to know is, are these regions present in infants? The trouble is, it's nearly impossible to scan infants. However, My amazing colleagues, Rebecca Sachs, shown here, her then graduate student, Ben Dean, and her heroic main subject, starting on her own son, Arthur, figured out how to scan infants and get sensible data. And this was really quite a feat. And so what they did is, I'll show you what's involved in scanning infants in a moment, but what they did then was to show nice child-friendly videos of faces and scenes and bodies to infants in the scanner while they were awake, looking at the images, and we collected functional MRI data when they did that. So we made nice, friendly videos like this. Those are kind of face videos. Here's a body video. Here's an object video and a scene video. And so we had many of these, and the kids go in the scanner and look at these images while we scan their brains. So you may be wondering, how do you get an infant into the scanner? So let me show you Rebecca placing an infant in the scanner and how this works. So there's the infant in a specialized coil ready to go into the functional MRI scanner. And here's the intrepid Rebecca doing what it takes to keep that kid happy and in the scanner. So she goes right in there with the kid. This is her son, Percy. Look at Percy kicking. Now you see why it is so hard to scan infants. Okay. Nonetheless, Rebecca and Ben and Arthur and Percy and a few other kids managed to do this study. And so here is the some functional MRI images of the brain of one 6-month-old infant, showing regions that respond more to faces. That's approximately the fusiform face area, and regions that respond more to scenes. Those are the regions in blue right there. And that was pretty heroic. But then, and it shows that the layout of these regions or approximate location in the brain are already in place by 6 months. But this study didn't really tell us how selective those regions are. And that's because it's really hard to get high quality data from squirming, kicking infants who don't stay in the scanner very long. So enter the scene, Heather Kosakowski, who got her PhD recently, jointly mentored by me and Rebecca Sachs. And I should say all of this infant work, the real heroic work, was done by Heather and Rebecca much more than me. They were very kind to let me kibitz and come along for the ride. So Heather set out to get better data from infants. And so first she worked with some physicists to design a new infant imaging system. And that involved designing new earphones with sound protection for the infants that you see here on the infant's ears, and developing a new scanning coil that fits snugly around the infant's head. And so you see it's open here, and then the infant gets comfortable, and then you put the coil in together. So so it fits snugly around the infant's head, giving nice high signal to noise around the brain. So then, even more amazingly, Heather scanned 83 infants. This is really nothing short of heroic. And let me show you what that looks like. So she started with 86 infants recruited. 83 of them went into the scanner, and from that, she was able to get at least a little bit of data from 74 of the infants. That's 49 hours of potential data from 324 hours of original scan time. You really lose a lot of data in this process. And here's why. Here are all 74 infants, and this is the amount of functional MRI data shown on the y-axis for each of these infants. So what is the problem here? Well, here is one infant showing you the amount of head motion over the period of the scan. And it's probably hard to read these numbers, but this kid was moving 9 millimeters here. That's a lot of head motion. And so you can't really analyze those data. And so to analyze these data, Rebecca and Heather developed a whole new data analysis method in which they look through the data for the subsets of the data in which the kids are not moving too much. Those are called subruns, and they isolate just those quality data. And that's shown in the orange bars here. And you can see it's a lot of the data do not get included in this final analysis. Okay, so these are the data that are going to be analyzed. Here is just 39 kids who have enough data here with low motion. So first, what Heather did is a fairly standard group analysis where she aligned those 39 infants' brains as well as possible and did a standard functional MRI analysis looking for regions that responded more to faces and objects, scenes and or places and objects, and body parts and objects. And what you see in this group analysis is something that looks a lot like a normal face, fusiform face area, parahippocampal place area, and extrastriate body area. Now, I hasten to add that these are group analyses, and this is at a very liberal statistical threshold. So this is sort of encouraging, but it doesn't really nail the case yet. So then Heather went on to do a kind of analysis that has mostly only been done in adults before, where you split your data into two sets, and you use one set to find the relevant voxels, the relevant 3D pixels in that brain, in that infant, where the action seems to be with this contrast. And then once you found those voxels, then you analyze the data in the remaining held out data to get an unbiased measure of the response magnitude in that region. And when Heather did this on her 39 infants, she indeed found beautiful face selective responses. So this shows you in independent data averaged over those 39 infants, the response in purple when the infants were looking at faces and the responses when they were looking at bodies and objects and scenes. And it was significantly higher to faces than to each of the other three conditions. So that's nice face selective responses in infants averaging only 5 months of age. For the place area, she found significantly higher responses to places than to faces, bodies, and objects. But it's negative because the baseline condition, which in this case was a kind of swirly, like a screensaver kind of image, was also quite high. So that doesn't exactly fit with the adult data, but the other properties of this response are very much like adult place selective responses. And in the extrastriate body area, she also finds body selective responses. That's the pink bar here, response to bodies compared to the other three conditions. So what she's found here is face, place, and body selective areas in 6-month-old infants. So much for the idea that these regions take many years to develop. What about the idea that they require visual experience with the relevant category? Well, my postdoc Ratan Murthy decided to take another whack at this question, and he decided to do that by scanning congenitally blind adults as they touched 3D printed objects like these. So here are some of the stimuli that he 3D printed. And here's his high-tech scanning procedure. So here's the participant in the functional MRI tube with a cardboard disc with these little 3D printed plastic objects velcroed onto the disc, with a subject standing outside the scanner, rotating the disc every few seconds so the blind participant can just feel those objects as they go by. And so first, here are data from the control sighted subjects looking at these images here. And this shows you the face selective response, higher response when you look at those images than when you look at these three other kinds of images in the fusiform face area, as we've seen before. The question is, what do you see in congenitally blind subjects who have never seen when they're just touching these images? And what you see is face selective responses in more or less the same location in blind participants with a similar kind of selectivity ratio of the response to faces versus non-faces. I will note that conveniently, I've made these numbers so small you probably can't see, but the absolute magnitude of response is substantially lower in the blind participants than the sighted participants. So it's not exactly the same kind of response. Nonetheless, it's a face selective response in that same region in the blind subjects. Okay, so do you need to see faces to develop face selective areas? Apparently not. Okay, so I started with these three questions. I asked how early do these regions arise in development? And I showed you that face, place, and body selective regions arise by 6 months of age. We asked about the role of visual experience for the face areas. And I argue that visual experience with faces isn't necessary for face selectivity in the fusiform gyrus. But I want to hasten to add that these answers are not true for all of these regions. In particular, this visual word form area that I talked about before that responds selectively to visually presented words and letter strings, it does not show up until kids are age 6 or 7 years. So much later, and in fact, it doesn't show up until kids are taught to read. So only kids who have learned to read show this selective response. So it's quite different from the other regions in the sense that they need not only lots of experience, but formal education with reading to develop this particular selective region. But that also gives us an opportunity. Because the visual word form area arises relatively late in development, that means we can ask, how does it know where to go? Because we can scan kids before they learn to read when it's not selective for visually presented words, and then we can scan them later. And we can ask what predicts where exactly it's going to arise in any one individual subject. So my long ago postdoc, Zainab Sagan, now a faculty member at Ohio State University, used diffusion tensor imaging, where you get these lovely tracks of long-range connectivity in the brain. And what she showed is that you can predict exactly where that region will arise in a 7-year-old after they learn to read by looking at the connectivity of that region at age 5, before they learn to read and before it's selective for words. And that suggests that long-range connectivity of each region of the cortex may be important in determining where things land. And so I'll just end with a few open questions. One, what about the development of the rest of the cortex? And I will just mention that in currently ongoing work, the ever amazing Heather Kosikowski has been studying auditory perception, and she's finding what's starting to look like a music selective response in 1-month-old infants. Here's a response when they're listening to music. Pink is speech, and the other bars are scrambled stimuli matched to the music with various properties. Of course, we want to know more generally what you have to build into a system to get these category selective regions to arise. And since I'm running short of time, I will just mention that my former postdoc, Katerina Dobbs, has been using machine learning methods to ask in a convolutional neural network, one of these deep nets, if you train it on face and object recognition, it spontaneously segregates itself into 2 systems, one for face recognition and one for object recognition, even though nothing is built into it to say, make separate systems for faces and objects. It figures that out without any priors. So of course, we don't know whether the training would correspond more closely to human evolution or individual lifespan development. And in any case, these networks got a lot of face input. So maybe what you have to build in innately in humans is some kind of bias to look at faces. And the final wide open question I'll just mention is that if indeed it's long-range connectivity of each region of the cortex that determines which functions will arise at each location, then that just pushes the question back one step. How does that long-range connectivity arise in development? And I'll end there and take questions.
Great, thank you so much. What an interesting presentation. I also very much appreciated the visual on how to do the MRIs on infants. That's a different aspect of measurement that many of us probably have not thought about. This presentation is open for questions, so I encourage people to go ahead and add their questions to the Q&A, or if any of the panelists have a question, to go ahead and raise their hand and share. I will start off with a question. This is not my area of expertise, but this work is really fascinating. And I found it just very interesting how you were able to really pinpoint the development of some of these regions much earlier than expected. In thinking about the implications for infant care and development and the priority placed on that richness of the early environment, how does this work inform some of that early infant, implications?
Yeah, that's a great question. I think this work doesn't directly inform it, but it raises questions, right? So there's a lot of work, principally in research on animals, looking at critical periods. And the idea of a critical period is the idea that you need to have the relevant experience in some particular window of development in order to be able to use it. And so we don't know if that's true here, but the very fact that these regions arise so early raises the question of what would happen if your visual experience was different in that window. And in fact, there's a little bit of data on this. There are studies that a number of groups have been doing on kids who are born with bilateral congenital cataracts, right? Of course, we can't control experience in young infants. That would be unethical. But we can do research on people who have different kinds of experience early on in development. And kids born with dense bilateral cataracts, if they're born in developed nations, usually surgery is done very early in the first few months of life. And those kids go, where you replace the lenses and give them not quite 20/20 vision, but very good vision. And so from then on, they have normal visual experience. So those kids are mostly fine visually as adults, although interestingly, there are very, very subtle differences in the way they process faces, even as adults 20 years after the surgery, just from having had, just from basically starting development a few months later than normal. So that suggests that there's not exactly a critical period, but a period when early experience is particularly important, at least for faces.
Great, thank you so much. And we have a really interesting question that has come in through the question and answer box from our audience. Some children learn to read before age 7, as early as age 3 or 4. Is it possible that the connectivity to the VWFA is set up even earlier? From an evolutionary perspective, how do you explain a functional area when writing has only been developed in the past 10,000 years? Big questions.
2 very good questions. So I don't know if it's standard, but there's an emerging view that most of the long-range connectivity of the brain is actually present at birth. And hence the idea that it can serve as this kind of framework within which functional regions develop. It basically pipes the relevant information to the right place, and functional development can happen from there. That's very far from proven, but that is sort of the idea that a lot of that long-range connectivity is present at birth, in which case that should work fine for kids who learn to read at age 3 or 4. I don't know that anybody has studied them with functional MRI. But your other question is a great one. What is the brain doing with specific connectivity to a region that's going to become a visual word form area when reading is such a recent cultural invention? In fact, like very, very few people learned to read until, just 3,000 or 4,000 years ago. And that's one of the reasons that I think the visual word form area is so interesting, is that we basically know that region can't be innately specified because reading is just too recent a cultural invention. So what is that connectivity doing? Great question, big puzzle. The methods for looking at connectivity in humans is not all that great. We have resting functional correlations and we have diffusion tractography, and that's kind of it in vivo. And neither of them is perfect. But what Zeynep Sagan has shown is that the visual word form area seems to be, and the connectivity of that region in 5-year-olds that is predictive of where the visual word form area will land is connected to the language regions. And so why would language regions have privileged input from high-level visual cortex? This is a fascinating puzzle, and we don't exactly know. It may be that before people learn to read, those regions may have played a more general social role in perceiving faces and other visual information that's important for understanding language. But it's a major puzzle.
Thank you so much. Well, it sounds like there's a lot of interesting research to come in this area. And I would just like to thank all of the speakers in this session. I think this really exemplified some incredibly novel and innovative work that's being done across many different spaces of measurement for behavioral and social sciences. So thank you so much to everyone for sharing this exciting work today. We are coming to a close, and I am going to turn this back over to Dr. Blackman-Demner, who is going to give our closing remarks for the festival.
Okay, great. Thank you so much. Can everyone hear me okay? Yes, I'm gonna assume yes. Okay, I just want to take a minute more of your time. I want to take this final opportunity to thank all of our speakers over the last 2 days, all of our moderators, the planning committee, and our wonderful meeting support staff. For all of their efforts in making this just a really fabulous and engaging 2 afternoons of presentations and discussions. A huge thank you to everyone for joining us over these last 2 days and for hanging in there till 4:30 on a Friday afternoon. We really appreciate it. Just a few brief announcements before I close us out. Please save the date for future OBSSR events. Our 2023 OBSSR Directors webinars will be held on March 28th, May 16th, and September 19th. Our 16th annual Matilda White Riley Honors will be held on May 22nd. And you don't have to write all these dates down. If you go to our website, subscribe to our listserv, you can find all that information there, and you'll get updates on all of the upcoming events, as well as BSSR news. And again, I just want to say thank you to everyone. I hope you all have a wonderful weekend and a happy holiday season. Thank you.